My name's Fornito. I'm from Monash University in Melbourne, and it's a real pleasure to kick off the first session of what I'm sure is going to be a fantastic meeting in the wonderful city of Rome. So hopefully you all found the place without too much trouble. And really the goal of today's workshop is to try to give you a reasonably comprehensive introductory overview to the field of connectomics and network neuroscience. And so we're going to be talking about issues related to how you go about building connectomes, and then what you do once you have those, how you go about analysing them. And so my talk initially is really just going to be an introduction to some of the basic concepts and some of the things to think about. And then the rest of the talks throughout the day are going to be focusing in more detail on specific aspects of network construction and analysis. Before we get started, OHBM is using a specific online platform that allows for more audience interaction. And so this will be particularly useful for anyone who wants to ask a question but maybe doesn't feel confident coming up to the microphones. So during any presentation, you can log into this website here, gmp3.cnf.io, or alternatively, if you've got the OHBM app, there's a button on the homepage. I forget what it is now. Let me check what it is. It is audience response, so third row on the far right. And if you go to either of those, you'll see all the sessions listed for the day. Our session should be the first one, the Network Neuroscience session. You click on that, you'll see all the speaker names. You can click on a speaker name, and then you can type in a question. And so then the moderators, myself and Andrew, will see those questions at the end of each session, and people can vote them up or down, and we'll try to ask as many of those as time allows. In addition, some speakers will have some interactive polls embedded within their talks as well, and you'll be able to see the live results. So please do make use of that. It's a good way, again, if you're not feeling confident in getting up and asking questions, it's a good way to get your questions answered. So, as I mentioned, the more sophisticated of us have embedded live polls into their talks, but being a Luddite, I wasn't able to get it to work. So I just thought I'd start with a quick survey, the old-fashioned way, using a show of hands. So the question is, I really just want to get an impression of how many of you, or what the levels of experience are in the room. So if I was to ask you how much experience do you have with network neuroscience, can I get a show of hands? How many people have no idea what a network is? All right, a couple. What about, I've never performed a network analysis, but I know what a network is? Okay, decent proportion. A little bit of experience, but not really confident. Okay. I know networks, so maybe you come from a background in math or physics, but you don't know how they apply to neuroscience. Okay. And you use it all the time, and you're a regular network neuroscience junkie. All right. So a bit of a mix, which is great. Hopefully there'll be something in this for all of you. As I said, it is pitched at an introductory level, but I guess, hopefully people learning the applications to neuroscience will get to see some of the idiosyncrasies with which networks can be applied, and obviously for people that are completely new, they'll get an understanding of the basic concepts. So first things first, we want to define our terms, and you've probably heard this word a lot if you're at this conference, connectome. So the term was first defined by Olaf Sporns, Giulio Tononi and Rolf Kotter in 2005 in this PLOS computational biology paper, and they defined it as a comprehensive structural description of the network of elements and connections forming the human brain. So there's two things to emphasize in this definition. So one is a structural description, so we're really talking about anatomical connections between brain regions, so essentially a wiring diagram of the brain. And although people use connectome in reference to functional connectivity, there could be an argument to be made that in a very specific sense of what might be meant by an ohm in terms of a comprehensive map or something, you could really only do that anatomically because functional dynamics vary so much. So in the strictest sense, it's referring to a wiring diagram of the brain, but people have adapted it and used it for functional connectivity. Now, the other key thing of this definition is the emphasis on a network description. Right? And this is key because if you think about it, we can find networks at all different levels of organisation in the brain. So we can map networks of molecular interactions that drive synaptic signalling. We can map networks of individual neurons and synapses at the microscale. We can map networks of cell populations linked by axonal fibre bundles at the mesoscale. And at the macro scales that we're typically used to looking at with, say, human brain imaging, we can map large-scale white matter fibre bundles and how they connect broader brain regions on the scales of millimetres to centimetres. Now, lots of people in the broader endeavour of neuroscience are doing work at each of these scales, and it all seems very specialised and complex, but despite the superficial differences between the scales, the measurement techniques, the model systems that are being studied, this kind of core theme of having a network description is common across all of these. And so the nice thing about network neuroscience, and in particular the maths on which it's based, which is graph theory, is that it provides us with a unified framework for representing these diverse kinds of neuroscience data. And so we can do that by representing the network in the most abstract sense or way, which is simply as a graph. Right? And so a graph is just a collection of nodes linked by edges. And so the nodes represent some kind of fundamental processing unit of the system. So if it's a social network, it would be a person. If it's a brain network, it could be an individual neuron or a specific population of cells. And the edges map the interactions between nodes. So in the brain, it could be structural or functional, depending on how we've measured it, and we'll go into that. And just to kind of illustrate how general this approach is, graphs have been used to model lots of different networks found throughout nature. So you can model networks of protein interactions within a cell, social networks of, say, academic collaborations across the world, or Twitter networks or Facebook networks, technological networks such as the World Wide Web or the Internet, and man-made networks such as infrastructure or transportation systems. So it's a remarkably flexible tool. So then the question is, okay, well, how do I go about building a graph model of the brain? And so the first thing we need to do is obviously define our nodes and edges. Now, in certain applications, defining a node can be relatively unambiguous. Say you're looking at the nematode worm, C. elegans, and you've got connectivity mapped at each and every neuron, then each node would represent a distinct neuron. But in the macro scales that we're used to dealing with MRI, the precise way in which to define a node is a little bit less clear. We don't really have any gold standards. And so we typically have to use a number of different heuristics. And so as a general rule, we would like our nodes to be spatially constrained. And so that just refers to the idea that we would think of a functionally specialized patch of the brain to be a kind of anatomically contiguous, confined space. We want those nodes to be intrinsically homogeneous, so everything that constitutes a node should serve some common function. So if we're doing brain imaging, if we want to define a set of voxels as a node, ideally those voxels would serve some kind of common function. And we want them to be extrinsically distinct, so the functions of one node should be distinct from the functions of another node. Right? Now there's lots of different ways of trying to meet these criteria to different criteria to different extents. So the classic example is Brodmann's cytoarchitectonic maps, and these have been translated into stereotaxic space in a probabilistic sense for some regions, but not all. But alternatively, you could use variations in chemoarchitecture, myeloarchitecture, sulcal and gyral boundaries. People parcellate the brain at random. We can generate functional regions of interest. There's a lot of interest in the moment at data-driven parcellations, so trying to cluster voxels based on similarity in functional connectivity profiles or other metrics. We could treat individual voxels as distinct nodes, or more recently, the Human Connectome project released their version of a new cortical parcellation based on a multimodal integration of different kinds of imaging information. And so later on today, Janine Bijsterbosch will talk in a bit more detail about different ways of defining nodes and some of the things that you need to consider. For now, I just want to say there's lots of different ways. There's pros and cons to each of those methods. And typically you'd want to tailor it to the question that you're interested in. And usually, depending on what you're doing, it can be good practice to try to replicate your results using a couple of different parcellations just to make sure your results aren't dependent on the idiosyncrasies of whatever method you've used. But assuming you're happy with your node definitions, we then want to measure connectivity between those nodes. And so when we're talking about brain connectivity, there's three broad classes of connectivity. So one is structural connectivity, which we've talked about. This is the anatomical or physical connections between brain regions. Now this is intrinsically directed, right? So each axon has a source and a target. But typically with MRI, we can't resolve that directionality. So for example, if you use diffusion MRI, we don't know where the connection begins and ends, so we can just map where the connections are. So this is a limitation. If you're using more invasive techniques like track tracing, then you can infer directionality. Then there's functional connectivity. So here we're talking about brain activity, and this is defined as a statistical dependence between physiological signals that we acquire in each of our different nodes. So we record activity fluctuations over time in each node, and then we compute some measure of coupling between those fluctuations, most commonly using a correlation, but there's lots of other different measures out there. Now this can be either directed or undirected, depending on the metric you use. And the critical thing here is that it's estimated at the level of the actual measured signals, so the signals that we measured. And this is an important distinction from the third class of connectivity, which is effective connectivity. So this is the influence that one neuronal system exerts over another. And here the key emphasis is neuronal system, right? So we're trying to make inferences at the level of actual neuronal dynamics. And so that means that if you're using an indirect measure of brain activity, such as fMRI, then you need some mapping between that measured signal and the underlying neural dynamics that generated it. So the classic example in the imaging context is dynamic causal modeling, or DCM, where you fit different models of causal interactions between neural populations, and then you convert those using a model into hemodynamic signals, and then you examine the fit between those estimated hemodynamic signals and your actual measured signals. Right. So they're the three broad classes. Now most studies focus on the first two, structural and functional connectivity, in network neuroscience at least, and so that's what I'll talk about. Now there's lots of different measurement techniques for looking at different aspects of anatomical and functional connectivity. Anatomically, we can use electron microscopy, track tracing, informatic approaches, diffusion MRI, and so on. Functionally, we can use invasive electrophysiology, calcium imaging, surface electrodes implanted surgically, MEG and EEG, or fMRI. But again, in the human context, the most popular tools are really diffusion MRI and fMRI, so I'm just going to talk a little bit about that today. So we'll start with diffusion MRI. And I'm sure most of you are familiar with the basic principles, but the idea is diffusion MRI measures water diffusion in the brain. Axons act as barriers to that water diffusion, so the water diffuses preferentially along the main axis of the axon. And so we can use that information to track the putative pathways of different axonal fiber bundles. Now there's lots of different algorithms for doing that tracking. Broadly, they can be split into deterministic and probabilistic. And so we do the tracking across the whole brain, and we can generate these maps of whole brain white matter. And then we use that to inform our connectivity estimates. Now, the specific way in which we process the data and the algorithms we use will affect the final results that we get. Right? And in general, there's a trade-off between the specificity and sensitivity of a tractography algorithm. So as a kind of rough rule, probabilistic algorithms tend to be quite sensitive, but also prone to false positives, whereas deterministic algorithms tend to be more conservative, so less prone to false positives, but prone to false negatives. And so it's just important to keep in mind that the specific algorithm you use to map the tracks is going to affect the final network that you get at the end. And Rob is going to go into more detail about some of the issues associated with processing diffusion MRI later this morning. Once we've mapped our tracks, then we need to estimate connectivity in some way, and most commonly connectivity between a pair of nodes is estimated typically as the number of streamlines that we reconstruct that pass through two nodes, or some measure of tracked microstructure averaged across the extent of the track, say for example fractional anisotropy. Now, it's important to remember, these are pretty indirect measures of anatomical connectivity. So the streamlines we reconstruct are not the same as axons. They're kind of rough models of the trajectory of an axonal bundle. And these measures are susceptible to lots of things like head motion, crossing fibers, complex fiber architecture, various sources of noise, and so on. So we need to be a bit mindful about the way we interpret these measures. And I won't go into the details, but for anyone who's interested in this work, I highly recommend this paper by Derek Jones and colleagues, where they talk about all of these different issues and limits on the interpretation of diffusion MRI measures of connectivity. There is a whole area of work that is devoted to trying to develop more sophisticated models of the diffusion signal that then results in more physiologically interpretable measures of structural connectivity. Rob Smith will talk a little bit about some of that later today. So again, anyone who's interested can check out some of these papers. So now I'll move on to functional connectivity. So here we're concerned with measures of brain activity. And so as I said, for each brain region, we record activity fluctuations over time. So we have a time series for each node, and then we compute some measure of coupling between those time series for every pair of nodes. So this could be with a correlation, a partial correlation, mutual information, any type of coupling metric that you might think of. So we do that for every pair of nodes, and then we generate a graph-based representation of the network. Now, because we're measuring brain activity, we need to think about the state under which we've recorded that activity. So by far the most common technique is to use what are called resting state designs. So someone lies quietly in the scanner, not performing any specific task, and we just record how the signal fluctuates over time. And generally it's assumed, rightly or wrongly, that that's a fairly stable psychological state. So then you summarize that activity over time with a single value, a correlation coefficient, and that's your final network. Over the last few years, there's been a lot of interest in looking more at dynamic aspects of activity. So in this case, you might do a sliding window analysis. So you construct networks at different snapshots in time, and that allows you to examine how the network evolves through the time period. And then finally, we could measure functional connectivity during a task. Now, the key thing here is that a task is going to drive brain activity in specific ways. And typically, we want to isolate the task-dependent effects of functional connectivity from more background, task, unrelated, intrinsic or spontaneous dynamics. And so, I've cited some of our own work there, but Michael Cole earlier this year published a really detailed consideration of some of these issues. So anyone thinking about doing task-related functional connectivity, I recommend that you check out his article. Now, one thing that I do want to point out is that pre-processing, as with diffusion MRI, also affects your functional connectivity measures. So fMRI, we do lots of processing to the data to try to reduce various sources of noise and improve signal. And so, we acquire data from the scanner, we pass it through some pre-processing pipeline, and then we get our process time series, which then form the basis for our analysis. Now, what actually happens in this box, the kind of meat that goes into the sausage, is quite important and can have an impact on your final results. So these are the results of an analysis we did last year where we looked at 14 different motion correction pipelines for fMRI. So lots of different ways of thinking about and trying to correct for motion. And so here I've just plotted the proportion of edges that show a residual correlation with motion after the denoising. And so you can see across these different pipelines there's quite a lot of variations. So using a simple regression of just 6 head motion parameters, nearly 70% of edges still have contamination by motion. If you use more sophisticated techniques, you can get that right down, particularly if you use something like global signal regression. That also does a good job. Now, I'm not going to advocate one way or another for global signal regression. For those that are interested, we've got a symposium about that on Tuesday, talking about new ways of thinking about the problem. But I just want to point out that the pre-processing steps that you apply will affect your final network measures. And it's not trivial, right? So if you want to go and do, say, a case control comparison. So here we compared people with controls, people with schizophrenia to healthy controls. Depending on how you've processed the data, you might see differences in some cases or no differences in other cases. And the nature of those differences is going to vary. So here on the left, we've got a map of connections where people with schizophrenia have got increased connectivity relative to controls. Here it's decreased. And the top row is one processing pipeline, the bottom is another. And you can see that there's quite a difference here. So here a lot of the changes are between hemispheres. Here they're more in the posterior of the brain. Up here they're a bit more anterior. Here they're more central. Right? And so the conclusions you draw from your analysis, which networks are affected, which specific connections are affected, are going to be dependent on the way in which you've pre-processed your data. So it's important to think about that carefully. Beyond that, assuming we're happy with our pre-processing pipeline, we then need to work out a way of quantifying functional connectivity. And Catie Chang is going to go into those issues in a bit more detail later today. But I just want to point out that depending on the measure we use, we can actually introduce some biases or non-trivial structure into the network that we need to be mindful of. And so we can show this with this simple example here. So up the top, I've got a simple network of 3 nodes, and these are the anatomical connections between them, right? So these top 2 nodes connect to the bottom, but they're not connected to each other. So it's an open triangle. In the bottom, we've got a closed triangle, right? So every pair of nodes has an anatomical connection to each other. Now, if I estimate functional connectivity of this network using a correlation coefficient, in the bottom case, I'll get something that corresponds well to anatomy. You'll get some non-zero correlation for every pair of edges. Whereas in the top case, I'm going to get a non-zero correlation for this top connection here, even though there's no actual anatomical connection. And this is just a mathematical property of the correlation coefficient. If A correlates with B and B correlates with C, then A and C have to be correlated as well. Right, so correlations will be sensitive to these kind of indirect polysynaptic connections. You can actually show, this is work done by Andrew Zalesky a few years ago, where just using the correlation coefficient imbues the network with a certain degree of structure. So if you just take random vectors, completely random numbers, with a length that's similar to the time series that we typically analyse, you generate networks from those, and you look at a property called clustering, which is essentially the number of closed triangles in the network. These networks have got a lot more clustering than they should, given the fact that the data were generated completely at random, right? Because of this triangle closure property. So you might say, okay, well, I'll just use partial correlation and that'll give me the direct interactions between every pair of nodes. But it's a little bit more complicated. In the case of the open triangle, you'll probably get something that corresponds with anatomy. But in the case of a closed triangle, it's a little bit harder and kind of depends on the dimensionality of the data. And so what we've actually found using the same sort of random number experiment is that these networks tend to be less clustered than they should be. So there's a bias operating in the other way. Now there's alternatives. There are regularized partial correlations that kind of have parameters allowed to kind of tune the degree to which you partial out indirect effects and so on. And there's obviously alternative measures of coupling. But the point of this slide is just to illustrate that certain coupling metrics can have their own specific properties that induce a certain structure into the network, and then you need to think carefully about the effects that that's having, and maybe that informs the type of null models you need to use for your inference. And Andrew Zalesky will talk a bit more about null models later today. But assuming we've navigated through all of those vexed issues and we're happy with our final pipeline and measurement techniques and everything, we're ready to build our network. So we've parcellated the brain into nodes, we've measured functional connectivity or structural connectivity for every pair of nodes, and then we can represent that in the form of an adjacency matrix or a connectivity matrix. And then from that matrix, we can build a graph-based model of the network. So I'm just going to talk a little bit more about this equivalence. So this is a kind of zoomed in version of a typical connectivity matrix. So here each row and each column is a different node, and each element encodes the type and strength of connectivity between every pair of nodes. So here, for example, the connectivity between node 1 and 2 is 0.8, between node 1 and 3 is 0.45, between 2 and 3 is 0.47, and so on. You can see along here the diagonal, we've got a series of ones, because this is just each node's connectivity with itself, and in this case this is a correlation matrix, so each node correlated with itself is 1. And so this diagonal can be used to split the matrix into an upper triangle, so everything above the diagonal, and a lower triangle, everything below the diagonal. Now, in an undirected network, such as one that we construct with correlations, correlations are undirected. A correlates with B, but we can't tell if A influences B or vice versa. The upper and lower triangles of the matrix are going to be symmetric, right? So the connection from 1 to 2.8 is the same as going from row 2 to 1.8. So more generally, we say that element A, ij, is equal to element j, ia. So the element in row I, column j, is the same as the element in row j, column I. In a directed network, say we've built a network based on track tracing and we can actually infer directionality. In that case, the upper and the lower triangle of the matrix are not going to be symmetric. You can have different information in those. It depends on how we've listed the connections. We could either list outgoing connections down the columns or along the rows, right? But in this case, it means that, say, the value in row 1, column 2 will not necessarily be the same as the value in row 2, column 1, all right? So that's how we encode directionality in the matrix. Now, most of the times when we're measuring connectivity, we have some estimate of weightedness, right? We're estimating variations in connection strength, and sometimes we'll have non-zero estimates for all values. So with a correlation coefficient, you'll get some non-zero value for all elements in the matrix. And so typically it's common to apply some threshold to the data to try to reduce the noisy low value estimates and just focus on the actual edges that are likely to be real. And often many people also binarize the networks. In this case, you set all connections that survive the threshold to the same value, value of 1. So by doing this, by binarizing, we're focusing just on where connections are in the network, ignoring their strength. Whereas if we keep the weights, we can look at how those variations in connection weights might affect network structure. Now, depending on how we've built the network, some approaches come with an intrinsic threshold already. So for example, if we build a diffusion MRI network using deterministic tractography, in this particular subject, we can reconstruct 28% of all possible connections. But if we apply probabilistic tractography to the same person, we get up to 60% of connections. Right? So this is just related to what I said earlier, that probabilistic tractography tends to be more sensitive, but also potentially some of these are false positives. Now, there's no single correct connection density in the brain. It actually depends on how you parcellate the brain. If you've got a parcellation of very large regions, the network is actually likely to be very dense because the probability of finding at least one axon between two large regions is going to be high. Whereas if you've got very smaller regions, the network will be sparser. We don't actually know what the estimates should be in a human. So generally people will apply a range of different thresholds to ensure that whatever findings they're trying to report are not driven by the choice of any specific threshold. Once we're happy with that, we then want to build our graph model. And so I just want to kind of show how a matrix and a graph representation how they're equivalent. So up the top, I've got a toy network here of 6 nodes, and down the bottom is the matrix representation. So you can see here, node A is connected to nodes E and C. So if we go to our matrix, we go to row A, we find a 1 for column C and a 1 for column E. And because this is undirected, it's symmetric. So if we go to column A, row C, row E, we see a 1 and 1 there. Right? So this is how we encode the connectivity of the graph in the matrix, and we can move back and forth between the graph and the matrix. We can do something similar with weighted networks. We represent strength of connectivity using the thickness of the edges, and in the matrix here with colour. So you can see here that the connection between C and F is the strongest connection, and if we go to our matrix, indeed, row C, column F, that's the strongest connection, or equivalently, row F, column C, because again, it's an undirected network, we get the same. Now with a directed network, you can automatically see that this matrix does not look symmetric, right? And so in this case, we've listed outgoing connections down the columns. So you can see here, node A has got 2 outgoing connections to E and C. So we go to column A, row C, row E, we see a 1. But then A's only got 1 incoming connection from node C. And so if we go to row A, column C, we get a 1, but there's nothing for column E because there's no connection going from E to A, right? So this kind of just illustrates how the matrix can be asymmetric, and that encodes the directionality of connectivity in the network. Now, different people use different connections. Some people list outgoing connections along the rows, and so when you're interpreting these in papers and so on, it's important to just understand how the connections have been annotated. And then we can do the same for weighted directed networks, right? So we can just encode or vary edge thickness to represent variations in connectivity strength. So then, given these variations, you might say, well, what graph should I use to represent the brain? Well, we can start with the simplest case, a binary network, so we just want to map where connections are, but we know that connections vary in terms of their strength over several orders of magnitude, according to some track tracing estimates. So we want to represent that. Maybe we want to use a weighted graph. As I mentioned, connections are generally directed in the brain, so ideally we'd use a directed graph. We also know that not all nodes are the same in the brain. So brain regions vary in terms of their intrinsic microcircuitry, their patterns of gene expression, their dynamics, and so on. And so we might want to account for that heterogeneity in some way. Now, graph theory provides us with a framework for doing that in the form of what are called coloured graphs, but it hasn't been used so much in the context of neuroscience. We can also account for heterogeneity in edges, right? So some connections might be inhibitory, some might be excitatory, so we can use signed networks, or we could account for other types of connections and so on. So really what we're talking about is ideally we'd have some kind of hybrid. We'd have a directed, weighted network that has heterogeneous nodes and edges. And this network would also evolve through time, right? So functionally, networks evolve from over scales of milliseconds to minutes, structurally over longer time scales. So we'd ideally try to represent that in some form, and Sarah Muldoon will talk about ways of doing that later today. So the nice thing is that graph theory provides us with the tools to be able to do this, but the reality is that most analyses so far are using the simplest possible model, and that's just a binary undirected graph. Increasingly, people are starting to look at weighted networks, and as our data improves, as we're starting to see these track tracing based connectomes, we're starting to see a lot more emphasis on directionality, and there's also been some work looking at heterogeneity as well, but it's been limited. And this is largely probably due to limitations in our measurement techniques. Things, especially with humans, things like directionality and heterogeneity are quite hard to resolve. So the nice thing is that graph theory provides us with a lot of scope to improve the sophistication of our models as our measurement techniques improve. Right, assuming we're happy with that, we've got our graph model, define our nodes, edges, we've got our matrix, we've got our graph model, we then want to analyse the network. And we can do that in one of 2 broad ways. So one is looking at the connectivity of the network, so this is focusing on variations in the strength and type of connectivity between nodes. And the other is looking at the topology of the network. And so the topology of the network really just refers to the pattern of pairwise connections in the network. So really you can think about it like the organizational principles of the network. So I guess more specifically, the topology of the network is any property of the network that doesn't change regardless of how you embed it in space, like if you rotate it or stretch it or expand it or shrink it. The basic configuration of connections between nodes will stay the same, and that's the topology of the network. And this is where we really use the mathematics of graph theory to obtain a wide array of different measures that quantify different aspects of network topology. And so really, as we move throughout the talks today, this is what we're going to hear more of in more detail. So next up will be Janine, who's going to be talking about issues related to defining network nodes and extracting time series for functional analyses. Then we're going to have 2 talks about defining edges. So Catie Chang is going to talk about measuring functional connectivity. Rob Smith is going to talk about measuring structural connectivity. And then there's going to be a block of talks that are focusing really on network analysis. So Andrew Zalesky will talk about statistical approaches to looking at connectivity and how you deal with multiple comparisons, and also appropriate null models to allow for inference on networks. Bratislav Misic will talk about different graph theoretic approaches to getting insight into communication processes on networks. Martin van den Heuvel will talk about how we go about defining brain network hubs and things like rich clubs and different measures of how integrated a network is. Rick Betzel's going to talk about modularity of brain networks. So there's community structures. So these are data-driven ways of trying to identify putative functional systems in the brain. And Sarah Muldoon's going to talk about multi-layer networks. So this is a kind of nice mathematical framework of linking different types of networks and analysing them in a succinct way. This is particularly useful when we're looking at dynamic networks, so how networks evolve through time. And then finally, Sarah Morgan is going to talk about applications. So once we've got all this knowledge, we've got all these different analytic techniques, we can build our connectomes. How then do we use it to address interesting neuroscientific questions? So hopefully today's program will give you a nice kind of bird's eye overview of the field and the key considerations that you need to think about. But just to summarise what I've been saying, so hopefully I've convinced you that graphs provide useful models of brain networks, and the real power is that it gives you a unified framework of thinking about networks across different scales, measurement techniques and species. There's different ways of defining brain nodes and edges and constructing networks. Each has pros and cons. You just need to be aware of how those pros and cons limit how you might interpret your findings. Brain networks can be directed or undirected, weighted or unweighted, and there's different ways of accounting for heterogeneity in terms of nodes and edges. So there's a lot of scope to build quite sophisticated models. And we can analyse brain networks at the level of connectivity or topology, and this can be done at the level of individual nodes or edges or globally across the entire brain. So I'll just close with some resources. So up the top here are some general texts on network science, so more from a kind of physics of networks perspective. The Newman and Barabasi textbooks are really good. These are some review articles. These are articles more specifically about network neuroscience, applications to the brain. These two books by Olaf Sporns were really kind of seminal works and a fantastic read. There's some software toolkits here that might be useful. A couple of years ago, Andrew Zalesky, Ed Bullmore and I published a textbook on brain network analysis, which is available through Amazon and Elsevier. And the field also has its own journal. It's been running for a couple of years now, called Network Neuroscience through MIT Press. It's open access and has very competitive publication fees, so if you're doing work in the area, please consider sending it our way. And I'll be down, there'll be an exhibition booth down in the sponsors section. I'll be down there tomorrow around lunchtime, so come and chat about it if you're interested. And my contact details are there if you've got any questions. But thanks for your attention. So, I forgot to connect to the internet. If you bear with me. Are there any live questions that anyone would want to ask while I'm doing that? Okay, you've got it? Good.
So the question is, in terms of correlation graphs, what are the implications of only including positive values, or would you use both positive and negative? So how to deal with negative weighted edges?
That's a very vexed question. So, the complication, if you're talking about fMRI, typically most of the negative correlations come about after you've used global signal regression. Other pre-processing techniques will give you some negative correlations, but they tend to be in the minority, whereas global signal regression will center the correlation distribution on zero, so you get roughly half negative, half positive. And there's been a lot of debate and controversy about whether those negative correlations are real. And one could argue, well, maybe it's distorting positive correlations as well. That's a kind of very long discussion. There'll be a whole symposium on it about it on Tuesday. I think if you do, if you say want to use global signal regression and you do get negative connections, then you need to account for that. You can't just, historically people have taken the absolute correlation. That might be okay for some applications, but negative and positive correlations are telling you different things about the interactions. And certain graph approaches, so for example, methods for community detection have been generalized to signed graphs. So the idea here is that if two nodes are positively correlated, they're more likely to be clustered into a community. If they're negatively correlated, they're likely to be in different communities, which makes a lot of sense. And a lot of other measures have been generalized to signed graphs, but not all of them. So depending on what you do, you might be a little bit constrained. But I do think it is absolutely worth thinking about those, and not just, say, excluding the negative edges or treating them as if they were positive. But there's no clear-cut answer. It's a bit of extra work to deal with them. Is that it? Great. All right. So next up, we have Janine Bijsterbosch, who will be talking to us in more detail about how we go about defining network nodes and issues related to extracting time series for functional connectivity analysis. Thanks, Janine.
Thank you very much, Alex, for that lovely introduction and for the invitation to come and speak at this interesting educational session today. So I am going to be primarily talking about how to define the nodes for network neuroscience analysis, a little bit towards the end about extracting edges. I should caveat this is a little bit more focused towards functional connectivity-based network neuroscience and a little bit less towards the structural side, mainly because these questions of defining nodes are particularly challenging, I think, on the functional side. So we saw some of these concepts already in the introductory talk today, and mainly what I'm going to focus on is how do we take the entire brain and how do we parcellate that into a number of brain regions, because typically we go on the assumption that functional regions extend beyond the size of just an individual voxel. And so what we're trying to do is define regions that are reduce the dimensionality of the brain as a whole into a smaller set of regions where each region is a functionally homogeneous region. And that's important because we're going to extract a single time series, and that single time series per region is going to be the input to all of the rest of our network neuroscience analyses, whichever method we decide to choose. So we're looking for regions that are homogeneous, and I'm going to talk about different methods in which we can try and define those. This is a sort of simple graphical summary of what Alex has just been introducing to us, of the sort of first steps that are shared across any network neuroscience analysis. And I'm going to talk about these first few here. And it might look like a little bit of a thankless task because, of course, typically what we're interested in is, the edges are the interesting things, and we want to model the connections, and we want to see how those connections might differ. But of course, it's extremely important to get the nodes right. If you don't define the nodes well initially, then anything that you do afterwards, whichever network neuroscience method you decide to choose, is basically going to end up, my GIF isn't working. That was supposed to be SpongeBob SquarePants running around and throwing something into the garbage, because essentially, if you don't get your node definition right, then anything you do afterwards is going to be extremely hard to interpret and essentially meaningless, because we need to make sure that the nodes and the time series are interpretable. Oh, there we go. It came up. Thank you. So let's talk more about node definition. And the first option that we have, and something that gets used, is to use what I'm referring to here as old school nodes, which are anatomical atlases. And some of the most commonly used ones here are things that you might have heard of, like the AAL and the Harvard-Oxford Atlas. And here, we've defined the nodes based on anatomical or structural information rather than functional information. And typically, because they're anatomically defined, they're based on a relatively small number of subjects. And this is the only slide where I really have a clear-cut message and a clear-cut type of advice of what to do, which is, don't do it. I think pretty much everyone in the field in functional network neuroscience agrees that these don't represent the functional organization in the human brain very well. So you can have a structural region that includes, for example, multiple different functional regions. And there's a lot of evidence for this. This paper here on the right is just showing some evidence in terms of predictions for behavioral information being much worse if you use these AAL and Oxford atlases, for example. So don't use anatomically derived atlases as your nodes. Okay, so we've decided that we need some sort of functional information to derive our nodes, and what I'm going to do for most of the rest of my talk is look at different methods that can be used to do that, and some different conceptual decisions that we have to make when deciding on how to define our nodes functionally. And the first option that we have, and one of the most common ones, is to do a data-driven clustering. And there are several different clustering algorithms available. The most famous one and the most best-known one is probably k-means clustering. I'm not going to go into all of these different methods for clustering, but if you're interested in it, I recommend the paper at the bottom, which compares some of these different clustering approaches. And essentially what they're trying to do, all of them, is they start off from some initialization, and then they optimize the clustering over time. And what this is trying to estimate is trying to assign each voxel to a specific cluster, so that you end up with clusters of voxels that are more similar to each other, or optimally similar to each other, and more different from the voxels in the other clusters. So you're not explicitly looking for the boundaries between nodes, but the boundaries occur implicitly because you're assigning the voxels. And there's a lot of different atlases available that have been derived in this way. I haven't put up all of the different references for these, because that would be a big slide in its own right, but hopefully you can find these when you do a quick Google search. So in the previous method we assigned the voxels to individual nodes, and the boundaries result implicitly as a result of that. But an alternative method that we can use is to use something called edge detection algorithms, where we're specifically trying to look for and define the boundaries between nodes. For example, you can, for every single voxel in the brain, look at its whole brain connectivity pattern, and then take the derivative or look at the change in that whole brain connectivity pattern across voxels. And that will give you something that you can see at the top figure here, which particularly looks for sharp changes in connectivity, whole brain connectivity patterns from one voxel to the next. And then you can use edge detection algorithms to draw lines in these sharp changes to define explicitly the boundaries between nodes. This is something that Alex mentioned already. This is one specific parcellation that I just want to spend one slide talking about. This is the one developed on the Human Connectome project by Matt Glasser and his colleagues, which is informed by functional information, both resting state functional information and also task-based functional information. But also this is a multimodal derived parcellation. So other information like folding patterns, myelin, cortical thickness patterns are taken into consideration. And this is actually a semi-automated parcellation. So some of the clustering and edge detection methods that I talked about already are fully automated, they're fully data-driven. Here there's some human labeling involved in deriving this parcellation. And the reason for doing that is to have it be strongly grounded and informed by neuroanatomy. So this is, if you're looking for a parcellation, this is one of the ones that you can have a look at. So now that I've talked a little bit, introduced a few of the different methods that we have available for defining nodes, I want to go over some considerations and some thought processes about what a node should look like, and how you should derive it. And one option is this idea of using either a data-driven parcellation on your own data versus a functional atlas in the literature. And the functional atlases that I mean here, they are data-driven. They are derived from data. They're just not derived from the data that you're going to use, the subjects that you're going to use for the rest of your network neuroscience analysis. So you have the option of either taking a data-driven analysis on a different data set from the literature, or actually running some of these algorithms on your own data and deriving your own nodes and running your own parcellation on your own data. And there's pros and cons to both of those. So if you have a particularly abnormal population that includes, for example, patients, then you would get a better representation potentially of your subjects by running a data-driven parcellation technique on your own data. However, it might be harder to interpret that and link that back up to the rest of the literature if there isn't a one-to-one mapping of the nodes onto parcellations that people might know and that are available in the literature. So which one of these is best for you depends on your research question and what you want to do. I also want to ask this question about should a node be a blob? And with a blob, I mean, should a node be a set of contiguous voxels that are spatially interconnected? And of course, having a contiguous node is the most intuitive and most easily interpretable, and it sort of fits with our sense of what a node should be like. But of course, the brain has bilateral homologs, so we know that these two regions are going to have quite similar time series in an fMRI scan. And so maybe they should, and they can be represented quite well with one homogeneous time series potentially. So maybe they should be combined together into a single node. And you can extend this further and say, well, we know that there's this network organization in the brain, and so we can define a node as a more spatially complex, spatially extended set of brain regions. And again, which one is the right option here depends on your research questions and the claims that you want to make, but there are these different options available. So far, I've talked about nodes as being binary, so nodes having voxels only having zeros or ones, and a voxel would have a 1 for one of the nodes that it's part of and a 0 for all of the other nodes that it's not contributing to. And again, that's the most intuitive for us idea of thinking about nodes. However, there is a case to be made for weighted nodes, for allowing voxels to potentially contribute to multiple nodes. We know that there's spatially overlapping network structure, and we know several people have published that spatial information, how that varies, how the spatial organization varies from one subject to another, is behaviorally relevant. And in fact, we've seen to some degree that if you get the node definition wrong, and if you don't accurately estimate the boundaries within a single subject, then that can influence the rest of your network neuroscience analysis. That's something that I've published on myself. And so there is instead of just having zeros and ones, maybe it's a good idea to have weights in each voxel such that if there's spatially overlapping information, voxels can potentially contribute to multiple nodes. And also, that allows us to capture some of the uncertainty of the node assignment. And an extreme example of that is gradients of organization, which I'll talk a little bit more about in the future. And again, I'm not trying to tell you that one is right or wrong. I'm trying to give you some intuition and some of the conceptual decisions that you have to make when defining nodes. And some might be more appropriate for certain research questions than others. So now, in the next 2 or 3 slides, I'm going to go over a few different methods, just like I discussed clustering and gradient edge detection methods. I'm going to go over a few different methods that we have available for doing a weighted parcellation. And one of them is independent components analysis, or ICA. And this is a data decomposition technique quite similar to PCA or factor analysis, if you're familiar with that, which essentially decomposes the brain into a number of nodes. But these are weighted nodes, and bilateral regions are typically considered within the same nodes, and depending on the dimensionality that you said, you might get more extended networks in ICA. Another example that's quite closely related to ICA is dictionary learning. So this is a linear decomposition technique, just like independent components analysis, but the optimization is a little bit different. So instead of optimizing for spatial independence, which is what ICA is trying to achieve, dictionary learning is trying to optimize to find sparse representations of the data. But again, it's going to result in a similar organization where you typically get more extended, spatially extended networks that are represented by weighted values in the voxels rather than zeros and ones. And the last option that I want to touch on briefly is Profumo. This is not as well known. It's an alternative to ICA, which does away with the independence constraint that you need to optimize independence components analysis. And Profumo is essentially a Bayesian framework, which has some nice features in that you can tell it what to expect, how spatial organization should look like, and have some other temporal priors in terms of the hemodynamic response function, and potentially even the connectomics that we see. But what's particularly nice about this framework is that it combines subject and subject-specific and group-level parcellation. So it iteratively optimizes between the group-level node definition and the subject-specific representation of that same node definition. And that's a nice feature, and I'll come back to that again in a few slides' time. I mentioned already one particular example of a weighted node representation is this idea of connectivity gradients. And this almost goes beyond the framework of having nodes and looking at connections between nodes, because what we say when we're talking about gradients is that in the same spatial location, you can have multiple directions, multiple dimensions of organizational properties. And we know that this is the case, for example, in the visual cortex, where depending on the feature of the visual stimulus that you're looking at, the neurons are organized and the neural populations are organized in different directions. And that has been extended to much of the rest of the brain, showing that there are these gradients, gradual changing gradients of connectivity patterns throughout the brain that sometimes overlap spatially. So I've mentioned this word a few times, and Alex mentioned this word a few times. I think it's about time to address the elephant in the room, which is dimensionality. So if we get this to work, this is a live poll. At least I'm hoping this is a live poll. Maybe this isn't going to, yeah. So you can now go onto your phones or onto your devices, and basically I want you to enter a number, any number. I think I allowed anything between sort of 1 and 5,000 or something. Of what do you think what's your favorite dimensionality for network neuroscience? Or if you haven't done network neuroscience before, what do you think is a good sort of number at which to represent the brain and study the brain? Oh, 17. Okay, that's a good point. That's a good starting point. Anyone else? Oh. It went up quite a lot, from 17 to 160, 150. Oh, this is fun. Let's see if we can get a stable estimate. Nobody's going anywhere near 5,000. That's interesting. But, okay. I think this is a running average of all of your responses that you're sending in. So it works, it updates, ooh, someone listened to me and just when I'll put in 5,000, because she said that it should be 5,000, it just jumped a lot. So this is really interesting, right? We've ranged from 17 all the way up to 300. And so we'll move on to the next slide if it will let me. Essentially, what I'll do first is give you some intuition about what dimensionality means in some of the different methods. So this is an example of how things break down in ICA, which is one of the weighted methods that we have, where in each case in the top left you see what the network looks like at a low dimensionality, so at a dimensionality of 10. And then in the rest of the map show how this same network gets split or decomposes into multiple networks as you, as the researcher, increase the dimensionality when you're running ICA. And there's two examples shown here, one at the top left, one at the bottom right. If you have Workbench View installed on your MATLAB, on your laptops, then you can go ahead and click on this link and you'll be able to download a scene file and you'll be able to interact with this a little bit more and look at a few other examples. So that's just to give you some intuition of what happens when you increase the dimensionality, which is a researcher set parameter for almost all of these parcellation techniques. And this is an example in ICA. This, of course, you see the same thing when you use clustering techniques. So this is an example of clustering techniques going from 7 to 17 to a much higher dimensionality. And of course, again, you can see how the higher dimensionality maps on to the lower dimensionality. And the main thing that I'm going to say about dimensionality is that there's no clear-cut best dimensionality. So I don't have the answer to the question that I asked earlier, and I don't think any of us really do. You often find consistent results across a number of different dimensionalities, which is a good thing, right? It means that your interesting findings are not driven by how you decided to decompose the brain and what dimensionality you decided to choose. And I think crucially, it's useful to realize that there probably is no answer to what is the best dimensionality to do network neuroscience, because that's just fundamentally not how the brain is organized. The brain is organized in terms of a complex hierarchy, and where you decide to cut off that hierarchy and define the nodes depends on what your question is. And so you can look at these big networks, or you can break them down into individual regions. And both of those are accurate representations of the underlying organization of the brain. So there is no such thing as one true dimensionality. One thing that I touched on a little bit already is individual spatial organization. So almost all of the node parcellation techniques define a parcellation or a set of nodes at the group level. And that's for good reason, because we want to have some correspondence. We want to be able to compare apples to apples. We want to be able to say this edge that we're comparing between node A and node B is the same edge between the same two regions across all of the subjects that we're studying. However, we do know that there's a lot of spatial variability even after doing common registration techniques. There's a lot of spatial variability between people, and we've seen this in a number of papers that have come out over the last years, and I'm just citing a few of them here. And so it's important to think about, once you've defined your nodes, how to account for that spatial variability. How are you going to map your group level node onto individual subjects? And there's a few options for this. One of them is to use a multi-layer classifier, which is particularly implemented in Matt Glasser's multimodal parcellation. One of them is to use dual regression, which is specifically designed for independent components analysis, and I'll talk a little bit more about that later on. Again, I want to give a little shout out to Profumo here, which has this nice framework where it optimizes both the group and the subject level at the same time. And so this tries to particularly do well at addressing this challenge of accounting for spatial variability between people. And of course, one thing that we might not consider very often when wanting to do network neuroscience is that we can use task-based localizers to identify the spatial organization within an individual subject. And this is not particularly practical if you want to do a whole brain analysis. But sometimes if you're only interested in a small set of regions in a particular system that you're wanting to look at, and there's a way to use localizers to define them, then that might well be a good option. So this slide is a little bit of a summary of all of the things that I've talked about so far, and a summary of sort of my thought processes about how to go about node definition. And one of the key challenges that we have to realize is that there's no ground truth. There's no way to really fundamentally, once and for all, answer the question, what is the best set of nodes to use in the brain? Because we just don't know. What we're working from is this dense connectome. So this is when we're acquiring our data, we have a certain number of voxels, and we can map out, we can treat each voxel as a node, which is typically not what we end up doing for our final analysis. But this is the starting point. This is the starting point of our node definition. And this is a big matrix of voxels by voxels, and what we're trying to do is reduce that dimensionality and somehow summarize that down into a smaller set of nodes. And I think you can think of different parcellation techniques across multiple dimensions. So first of all, I've explained on the vertical axis here, some parcellations are hard or binary. They just have zeros and ones, and some are soft node definitions, and they might have weights. But also there's a crucial question about how information is represented when we're trying to define our nodes. And so I'm going to put some of the different types that I've talked about so far on this continuum, and this is just my idea of where these sit in this sort of two-dimensional framework. We can have discussions. You might disagree where I've put your favorite type. But essentially, some of them at the top here are hard parcellation. Some of them are clearly soft or weighted parcellations. I've put AAL outside. It is a hard parcellation, but I would say it doesn't really represent information very well at all, and that's why I've put it outside of the graph. I've also put gradients outside of the graph because it is a soft parcellation, but it's kind of so overlapping that it violates some of the assumptions that we make when trying to do network-based analysis. And so it principally disagrees with the idea of even being able to define nodes at all if there's too much overlapping hierarchy. What I would say is that, in the green area here, that's probably where we want to be for network neuroscience. We want things to be represented more temporally. Whereas some decompositions, such as temporal ICA, there are no edges left. It explains all of the information in the dense connectome spatially in terms of how the networks are organized. And so you couldn't do a correlation analysis afterwards because the correlations are forced by the decomposition to be zero. So we want to be in this green area because we want information to be mapped out, organized, and represented temporally so that it is conductive to doing network neuroscience subsequently. I would also say that having a hard parcellation right at the top here, the red area, having a hard parcellation that contains a lot of spatial information, that probably doesn't exist. That's just not how hard parcellations are. They're not flexible enough to represent this. One thing I would say is that there's this area in the middle that is not explored much. There isn't much work in terms of looking at whether it's a good idea to combine hard and soft parcellations. And I think it might be interesting because maybe some areas, such as early primary and visual areas, are better represented as binary hard parcellations, and maybe some areas where you see more overlapping functional organizations, such as in the parietal cortex, might be better represented by weighted representations. And I think that's just a question that maybe the field might become more interested in coming years. So now that I've talked a lot about considerations and different methods for node definition, I want to just talk briefly about time series extraction. Because of course, the time series is the fundamental part of your functional-based network neuroscience analysis. This is what's going to drive the rest of your analysis. And there are multiple different ways of doing this. First of all, if you have a binary nodes of ones and zeros, you can just extract the mean across those time series, across all of the voxels. And that's what's done most commonly, and that's probably fine. That's probably, if you've done a good job at defining the nodes and accounting for the subject-specific spatial organization, then that's probably a fairly good representation, although there are some things to take into account. If you're averaging, then the number of voxels that you're averaging across are going to have an effect. You're going to get, the more you average, the more clean or the less noisy your time series are going to be. And so if you have bigger parcels, then you're going to have cleaner estimates of that time series, assuming that there's a homogeneous time series representing those voxels. You're going to have potentially some parcel size effects. And also, potentially, if you're just averaging, you're not taking into account any potential noise. So it might be that some voxels towards the boundary, towards the edge of your node, are already kind of overlapping a little bit, or you have misestimated the boundary a little bit, and they might represent a different functional region. And that might affect the average if you just take the simple average. And so one alternative option that was trying to address this issue of noise is to do PCA and take the first eigentime series, which is essentially a weighted average. So rather than taking the simple average, take some sort of weighted average in an attempt to downweigh some of this noise effects. However, what you find in practice is that you often get more than one time series that are bold-like, that have low frequency information in them, and so deciding which one to pick or how to combine them can be quite challenging. So you don't see this often in the literature anymore, but I just wanted to point this out as a sort of idea that is in principle an idea to overcome some of the noise effects, but in practice is maybe harder to apply. There's also, for weighted nodes, there are other options. So you can use dual regression for ICA maps, although they are, as I'll explain on the next slide, potentially biased in the presence of spatial overlap. And again, there is Perfumo, which is this holistic combined framework, and it actually estimates the time series for you as well. One thing to note about Perfumo, I know I've said good things about it, but actually you get quite weak correlations afterwards. And so potentially, perhaps it represents too much information spatially to be a good node definition methods, particularly for network neuroscience analysis. This is just a quick slide on some of my own work, and here I'm showing that I've listed the poster there as I've done on some other slides. So if you're interested, go ahead and go to the poster or the symposia that I've listed, and you can find out a lot more. Essentially, the idea is that if you have two networks which are represented in 2 dimensions here, and they're overlapping to some degree, then because ICA is optimizing for independence, it's going to estimate them fairly well, but it's going to underestimate some of the weights in the region of overlap, and it's also going to change the background a little bit, as you can see on the right. And this actually changes the correlations that you find. So this is a simulation, so we know the ground truth correlations. They're in blue on the top figure here. And actually what you get if you do dual regression, if you do ICA and then dual regression, you get a shift in those correlation patterns, which is introduced as a result of the overlapping, ground truth overlapping organization. And so we came up with an extended version of dual regression called thresholded dual regression. And again, if you want to hear more about that, then I'd invite you to come to my poster and have a chat with me there about that. So I'll wrap up, and I'd love to have a lot of discussion because I haven't really given you many specific answers or clear instructions about what to do, and that's because this field of node definition is still developing, and there are a lot of considerations, and what to do depends on the research question that you have and the data that you have, and how you want to be able to relate it to the literature. But to summarize some things, node definition is a crucial step in any network analysis. And so it's really valuable to spend some time. It's easy to get excited about your network neuroscience and sort of rush past this point and just use AAL and get on with it. And I'd really recommend that you spend a little bit more time, thinking about some of the considerations that I've talked about today, and making an informed decision on how to define your nodes. Parcellation is a hard problem because there is no ground truth. We can't just have a look at what it should be. It's hard to validate. Don't use AAL. And realize that whilst this question of dimensionality is a challenging one, there's probably not one single true dimensionality that fits the brain. And this idea of needing to account for individual spatial variability is, I think, something that the field has only fairly recently, only in the last few years, sort of really realized, and so I think there's a lot more methods that are going to come out for really robustly projecting group level nodes onto individual subjects. And so with that, I'll end. Those are different ways for getting in touch with me. And a quick shout out, we wrote a really short textbook about functional connectivity analysis in particular. Node-based, node definition is part of that, but there's also other parts in that you can have a look at. It's available on Amazon. So thank you very much, and I would welcome any questions.
Hi Janine,
Hi.
Thanks for your presentation. This is something I've been thinking about. I work a lot in development and trying to look at how these functional networks develop, particularly as cognition develops. And typically we would look at how an edge strength or whatever changes over that development. But something I've been thinking about recently is whether the nodes, the extent of the nodes actually changes, particularly sort of functionally defined. I don't know, I'm just interested in your thoughts, whether anyone's done this, looking at different nodes as development occurs.
Yeah, that's a great question. So typically in a network analysis or node-based analysis, we just assume that the nodes are fixed, and that's something that is sort of the foundation of spending time defining them well. And there are other methods outside of network neuroscience that are available to look at changes in functional organization over time. For example, ICA plus dual regression is a framework. If you look at it, if you use it at a lower dimensionality, you can also look at spatial changes because you get the spatial maps, and you can potentially do longitudinal effects. I've not seen any work actually. There's this sort of continuum of where information is represented and whether information is represented spatially or temporally is, I think, what this question maps onto. And you're saying, at the moment we're assuming and we're only looking at changes in the temporal representation, so in the edges, but maybe we should be somewhere in between and also consider some of the spatial changes. Fundamentally, of course, the dense connectome is the foundation of both the spatial and the temporal changes. And so depending on how you split that up and reduce the dimensionality in that, you will still, even if the nodes change, you might see that as changes in your edge strengths, for example, because if a node sort of grows and your boundaries don't quite map on very well anymore onto that node, then it's going to reduce in network strength, for example. So you're going to get that information, but perhaps a more meaningful or interpretable version of that information is to look at changes in the nodes. I've never seen a sort of hybrid analysis that allowed for the nodes to change and for the networks to change, personally.
Thanks for your thoughts.
So we have an online question. So in the graph, how come temporal ICA is on the spatial side of the x-axis and spatial ICA is on the temporal side?
That's a great question. That's kind of confusing. So what I was saying with the horizontal axis is where the information is represented at the end of it. And so when you do temporal ICA, you optimize for temporally independent components. And so by definition, your sort of connectivity matrix, your correlation matrix is going to be 0, because that is what you've implemented as a version of doing that node definition that way. And that means that all of the information about connectivity patterns, I mean, you haven't removed the connectivity patterns from the data. It means that you've chosen to represent those connectivity patterns in terms of the spatial maps. So essentially, you can think of, you know, if two regions are spatially connected, they have to be in the same map, because we can't represent any information temporally. We can't represent any correlation patterns. Whereas spatial ICA is sort of on the other extreme, where you optimize for spatial independence, and so things are not really allowed. Well, you can have some overlap, and that's a complicated story for another day. But you enforce the maps at the end of it to be spatially independent. So if you look at the correlations between the maps, they are forced to be 0. But because of that, you enforce more of the information. You push more of the connectivity information into the correlation matrix and therefore into the temporal domain.
And one more. How do you determine which PCA components of a ROI are relevant?
Does this refer, so I guess this refers to using PCA for time series extraction, which I think is not that common anymore. Typically, the way to decide whether things are relevant or irrelevant, or whether they're largely containing bold signal information, is based on the frequency spectrum, because what we measure in fMRI, in general, functional MRI, is all that we measure is through the hemodynamic response function. So even though neural activity happens at a very fast time scale, what we're measuring is sort of the body's hemodynamic response in sending extra blood to that region, and that happens over the time scale of several seconds. It's really quite slow, and therefore, because of that, by definition, any sort of neurally based information will have most of its power in the low frequency range, because we observe it sort of through the veil of the hemodynamic response function. So if you see PCA components or ICA components or anything like that that are sort of have a lot of high frequency information in that, typically the majority of that is noise derived rather than signal derived, because of this hemodynamic response function.
Great. Well, join me again in thanking Janine. It's fantastic. Thank you. I noticed one of the questions getting voted up is it possible to upload the slides? So the speakers will have uploaded their slides to the HBM website, so people that have registered for the educational workshop should be able to hop onto the HBM website and access the slides. Hopefully all the speakers have done that. So moving on now, next up I'll introduce Catie Chang from Vanderbilt University, and she's going to be talking to us about different issues related to measuring functional connectivity.
Thanks so much, Alex, and good morning to all of you. And today I wanted to focus on some techniques and considerations for quantifying functional connectivity. So I thought a good place to start might be to discuss a little more about what is functional connectivity. And so one common definition is that we're quantifying statistical associations between the time series of different brain areas, right? And so a motivation is that if areas are sharing similar activity time courses, then maybe there's some kind of direct or indirect interaction going on between them. But it's important to actually remember that here we're not looking directly at physical or structural connections between regions, and we're not really doing any sort of causal interventions to try to find out if these areas are actually interacting. This is more about statistical associations, and so it's just important to keep in mind when we go about trying to interpret our findings. So some approaches, actually many types of approaches for functional connectivity, emphasize the type of viewpoint that I'm showing here, which is that we're looking at associations between the observed time courses of different brain areas. But there are other types of methods, such as ICA, PCA, et cetera, that take a slightly different perspective on the data. And so in ICA, for example, we're taking our whole four-dimensional data set and decomposing or unmixing that into a set of underlying components or building blocks like these. And so another kind of perspective on functional connectivity is that areas that appear together in some of these components may also be considered to have functional connectivity. So I'm going to start with some common techniques for looking at functional connectivity on more of a voxel-wise whole brain level, so that's seed-based correlation and ICA. And then move on to, I think, what we usually think about a little more when we think of network neuroscience, which is in the relationship between a set of nodes. And then discuss some challenges in interpreting functional connectivity. And I'm going to be emphasizing fMRI data. So seed-based correlation is probably still one of the most popular methods that we use for functional connectivity, and was, I think, the very first approach from Bharat Biswal's original paper. And the idea is that we first choose some seed region of interest, and you extract its time course, and then we just ask how strongly correlated is this seed time course with every other voxel in the brain. And this can be represented as a 3D spatial map here, which is colored according to the strength of how each voxel correlates with that seed. And then typically we do some transformation from correlation to using a Fisher-Z transformation to make it a little more well-behaved for our statistics. And then we can subject these maps to some kinds of statistical tests, typically over a number of subjects. And so we get a seed-based functional connectivity map that's associated with where we placed our seed. And so if we place our seed in different regions, then we're going to pull out different correlation patterns with the rest of the brain. So as an example, if we put our seed in the PCC, then that tends to have strongest correlations with areas that we know as the default mode network. If we place it in a motor cortex or visual cortex, we'll get maps that actually pull out pretty well motor and visual systems. And that's actually capturing one of the key findings of resting state fMRI, which is that correlations in our data tend to really closely match well-known functional systems. And so once that was noticed, then correlation analysis became a really popular method for trying to look at the brain's functional organization. And so we can look at maps on these kinds of spatial scales, and we can also move down to even finer scales. And so this was an example looking within the visual cortex of seed-based analysis, seeded from areas with different eccentricity representations. And the seed-based correlation maps actually really well pull out these different these organization in the visual system, and that's even when people had their eyes closed. So this organization is really in the spontaneous brain activity. So as we've heard, in network analysis and also for the seed-based analysis, one of the main considerations is in the seed itself. So where are we going to place it? What size and shape is that going to be? And usually instead of a single voxel, we average over at least a few neighboring voxels and try in order to try to boost the signal-to-noise ratio. And so we've heard a lot about this, and it's a great talk by Janine, so I won't go into this in very much detail, but the choices will depend a little on exactly what you're studying or what your specific hypothesis is. And so you might use like functional areas from predefined atlases, or if you have a more specific idea of exactly what coordinate you're interested in, perhaps you'd use that, or more individualized techniques such as functional localizers. And the choice of seed region is very important because this can really impact the results of our analyses. And so as one example, the default mode network has been reported to subdivide into at least two subnetworks, such as the dorsal and ventral default mode network. And each of these have distinct functional connectivity patterns. And so if you're saying to yourself, I want to choose a seed in the default mode network, and you happen to place your seed within, say, ventral default mode network, then you'll pull out a slightly different correlation pattern than if you were to place it in like the dorsal default mode network. So that's just something to keep in mind here. And this is just another illustration of the sensitivity to seed location, as we've already heard a little bit about already. So this is looking at what happens if you basically move your seed region along this little line from two areas in parietal cortex. And so as you're pushing your seed along from region A to region B, you can trace out, and you're taking a seed-based correlation map at each location, then we can trace out a series of different seed-based maps. And you can see that they can even change rather abruptly, even with kind of small shifts in the seed. And on the one hand, that might be a little bit worrisome, because if you have, like, say, errors in your registration or alignment, then that might cause you to place the seed in a region where you may have not intended to. But on the other hand, this is also really interesting information, perhaps for drawing functional boundaries between different regions. So like with these gradient-based techniques, you can kind of see where the map changes abruptly, and that's perhaps a region where there's a functional boundary. And this is just a movie, if I can play it, yes, that is illustrating a similar concept. So if you move a seed region continuously across this part of the brain, and we can look at where the associated long-range correlation patterns, which morphs around really nicely. So I just really like this visualization. And so just to recap a couple of thoughts on seed-based correlation analysis. First, this is yielding a whole brain, if we want, spatial pattern of how correlated each voxel is to every other voxel. And so we might use this type of analysis if we want to ask questions like we want to screen for which other areas in the brain are correlated with a particular region, or if you're doing a group level analysis and you want to figure out how is the correlation with a particular region modulated across different populations, like where is that changing? And so it is straightforward to calculate, in a sense where we're just taking correlations. But some considerations include the sensitivity to the seed size placement, and also that if we're looking at just the observed bold signal in a voxel and using those to compute our correlations, that can be a mixture of several different underlying sources, not just artifacts and noise, but also different neural sources which aren't explicitly separated. And we now know this. We have this complex picture. There could be overlapping networks and so forth. And so seed-based analysis doesn't really explicitly account for that. So let's talk about ICA right now, which is another very popular technique. And just to give you a little cartoon of the basic idea, basically we input our fMRI data. So suppose this is our sequence of bold volumes at each time point across the scan. And then what we get is a set of, if we're doing spatial ICA, we get a set of components that linearly combine at each time point to generate the observed data. And so in other words, at every given time point in our fMRI data, we can model that as a mixture of the activity that's coming from these different components. So at time 1, we might get some coefficient A1 times this pattern plus A2 times component 2 and so forth. And in spatial ICA, these weights can change, or these weights actually change as we move across time, but the components, the spatial maps here are what remain fixed. And so these can kind of be thought about as our building blocks for generating our fMRI data. And then we can also think about each component as having an associated time course, which is defined by the weights of that component's contribution to our data over time. And so if we just consider the first one, we can see how strongly it might be contributing over time, which is tracing this out. And this whole schematic is just a kind of another way of intuition behind, I guess, the matrix decomposition picture that I showed earlier on in the talk. And one common analogy that's made for ICA is this cocktail party problem. So it's like, imagine that you're in a party or some crowded room and you're just hearing a mixture of different voices, and your goal is to separate that out. Your brain does a great job of that, and trying to unmix into what individual person is saying. And another point is that actually, with ICA, we don't know this, and we don't know this. And so we have to solve for those things, and we can try to make this tractable by placing assumptions here, like that these are spatially independent. And so it's kind of interesting that this is not looking for networks per se, but since this structure is in our data, it actually does a really good job of finding it. I mean, even this, we're just using this mathematical criteria, but we get these very biologically realistic looking networks, and a lot like the ones that we find in seed-based analysis. So these are some thresholded ICA components that come from running ICA. You can see the default mode network and other familiar networks. And in addition to components that are going to look like real large-scale networks, we always also get a bunch of different artifact-looking patterns that may not be of interest, like ones where you see bright things around the brain or maybe in the ventricles. And so ICA is actually a really nice way of visualizing the underlying structure of our data, not just for neural patterns, but potentially for identifying artifactual patterns, and maybe not ones that we would have suspected were there before. Maybe something weird happened during the scan, and we can see that with ICA. And so some approaches that are used are, well, you can choose one or the other to focus on in your analysis, or you can actually try to denoise your data by essentially choosing components that you think may be noise, and then trying to eliminate those and reconstruct a cleaner data set. But all of these things require decisions about which components you're actually going to be looking for, and we want to do that ideally in an objective way. And there have been several different approaches suggested for this. So as just a couple of examples, FIX uses basically machine learning techniques to have learned spatial and temporal signatures of artifact versus neural components. So then you can hopefully automatically classify these in your own data. And another technique that's becoming more popular, I think, is multi-echo ICA. But one thing to note is that this does only work on multi-echo data. So that means that you'll have collected your data where, I guess instead of the typical way of getting one image per TR, you instead acquire images at different echo times within a TR. And this uses the principle that bold signals, like real neural, or like neurally generated hemodynamic responses, these bold related signals show an echo time dependence scaling in their percent signal change. Whereas non-bold artifacts or non-bold components, like things like head motion, would not be expected to have an echo time dependence. And so you could essentially think of then going and ranking your components along a spectrum like this to try to see which ones might be likely neural, and hopefully the artifact ones kind of separate over into another territory. But there's always these tricky decisions, and some types of artifacts don't actually separate very well along these lines, and so certain effects that we might not be interested in, like slow changes in breathing or heart rate, like these are actually bold-related mechanisms, and so therefore we need other mechanisms or methods if we wanted to try to separate those. One more point is that ICA, in ICA, the group level analysis is not quite as straightforward, I think, as in correlation-based techniques. So if you think about what happens if you run ICA on individual subjects, there's actually no guarantee that you're going to get corresponding components, although there's ways of trying to look for those. And so one approach that's been used more often, I think, is to try to do some group level or concatenation of your data. So if in temporal concatenation, for example, you make this giant data set by concatenating things over time across your subjects, and then you try to find a common set of group ICA maps. And then there's ways of individualizing this and going back to the original data by, say, back projecting these, or using methods like dual regression to try to find then the individual subject expression of these patterns, which might be input to group level statistics. And then there's more and more techniques being developed for this kind of basic idea. So here I wanted to list some considerations for ICA, kind of alongside ones we've discussed already for seed-based analysis. And so seed-based analysis is a pairwise method, right? So we are choosing a seed and we're doing sort of a 1 to many mapping, a seed to each voxel. ICA, on the other hand, is a kind of full multivariate decomposition where we input our whole data set, and it's going to extract multiple networks at once. And on the one hand, that's kind of convenient because we don't need to choose our seed region specifically, but there are choices that we've already heard about that are going to influence the output we get from ICA, such as the dimensionality or how many components you want in your decomposition. There's sort of automated heuristics for trying to pull out what the optimum number of components might be, but still, it's going to what we get out is really going to depend on the granularity we're going to ask for. So if you imagine asking for 5,000 or 2,000 or 20,000 components, well, can't ask for that many usually, but it's going to split the data much more finely than if we ask for only 15 or 30, and so then you can see things like canonical networks splitting up and so forth, which could be good or bad depending on your goal. And ICA can also be used for denoising, as we just discussed, and the group level analysis. There's a lot of interesting work being done along these lines for looking at these. And then this article is an old article now, but very, it offers a lot of interesting points about the analysis and comparison between seed-based and ICA methods, if you're interested in reviewing more about that. So as I mentioned, these seed-based and ICA are done, these are more like kind of voxel-wise analyses. And now let's talk a little more about how we might model these associations between sets of nodes. And as we've heard already in Alex's introduction, actually I stole this from one of his previous OHBM talks that was online, we go through these kind of procedure for doing a network analysis. So it usually starts with defining our nodes, extracting the time courses in some way, and then we want to define edges, or the strength of interactions or associations between these brain areas. And then we probably want to do some further analysis, especially if we're looking at complex networks or whole brain networks. There's also techniques that focus on a smaller set of networks, and actually the inference for the network edges might be the final step, depending on if that's your goal. So methods like DCM or structural equation modeling, for example. So a little bit more on how we define edges. So I'd say correlation and partial correlation are two very widely used methods for quantifying these associations. Pearson correlation, I think, is the most common one that I've seen, and this is where you simply calculate the correlation coefficient between a set of nodes, or pairs of nodes, and we can represent that as a matrix like this. We actually get the full matrix, but since it's symmetric, it's fine to show just one triangle. And an advantage is that it's simple to compute, and it's straightforward to interpret in the sense that we have a good idea of, we have a good intuition about what correlations are, and what that means in terms of our observed signals. But on the other hand, it doesn't maybe distinguish between direct and indirect associations between the nodes. And just to pull up this cartoon that actually Alex described a little already, suppose you have 3 nodes, and we know that in reality there's only interactions from C to A and B, but nothing between A and B themselves. And so if we are looking at Pearson correlations, it may be likely that we'll also see a relationship between A and B, even though they're not directly related to each other, but only are being driven by the common influence of node C. But then, if you suppose we go and partial out or regress out the effect of C from both A and B, then this is likely to disappear unless there was already some relationship between these that existed independently of C's influence. And so that's a basic idea behind partial correlation. So we essentially get the correlation between 2 nodes after partialing out the influence of all of the other nodes. And in a sense, that might be more specific to direct interactions, but on the other hand, there's also a danger of removing accidentally interactions that were already there and getting false negatives. Oops. And also, what happens now if we scale to very large numbers of nodes? Well, then every time we want to compute a pairwise correlation, then we're going to partial out the influence of hundreds and hundreds of nodes from each of those time series. And so this is going to eat up a lot of degrees of freedom unless we have very long time series. And also imagine what happens if you have nodes that are highly correlated already that are in your analysis. This can make the estimates unstable just because of collinearity. So if you think about, if we have two very highly correlated nodes and you regress out the influence of one, then you've removed almost everything from that other time series. But actually, we might say maybe those nodes should have been actually merged, or those, into the one big node to begin with to avoid that. And these estimates can be a bit noisy, and it's been shown through a lot of literature that regularization techniques may help to improve the robustness of this, such as by assuming, by using like a sparsity. Another metric I'll mention is coherence, and for fMRI data, this tends to be used less frequently compared to correlations, but can be conceptualized as a frequency domain version of correlation. And since it's a function of frequency, then one thing that it allows us to do is essentially decompose the linear dependence into a specific set of frequencies, or frequency bands. And so just as a simple example, in one of the earliest techniques or papers to use this on fMRI data, if you just make a large band of the low frequency spectral content, so 0 to 0.15 hertz compared to higher frequencies, and you look at the relationship like a kind of seed-based coherence analysis with a voxel here, then you'll see a lot more here for the lower frequency band than the higher band by virtue of the fact that we have a lot more energy in our hemodynamic response in the lower frequencies. And another property is that it detects any linear relationship between signals. And so if you have two time series that are just shifted from each other, for example, which might pop up a lot in fMRI if we have hemodynamic response differences in different brain areas, a zero lag correlation might be sensitive to that time shift, but coherence wouldn't be. And in fact, it's more generally, if one time series is a filtered version of another time series, then it doesn't affect coherence, but definitely could affect correlation. And this is much more commonly used in electrophysiology than fMRI. One reason is probably because in electrophysiology we can see oscillations at certain frequency bands that have better known functional relevance. Whereas in fMRI, and especially when we're looking at resting state signals, it's a little less clear to us which frequency bands would be of interest, although a number of studies are now trying to explore that and get more insight about that. Anyway, but this metric could be of interest if you wanted to focus our network associations on particular frequencies of the signal. And these are just a few of many, many actually possible metrics we can use. And I talked about some linear metrics, but there's also nonlinear measures like mutual information that use higher order statistics and to quantify associations. And so one question that you might wonder at this point is, which metric should we use? And I think a common theme of the discussions today is that there's no right answer. And I think, for brain activity data, the question, it's always really hard because we don't have true knowledge as to what we're supposed to get, to get the best quantification of our network. But also, some of these metrics provide complementary information. So then maybe the question isn't about what's better, but about which type of information in our signals we want to focus on. Do we want to ask about just correlations without lags, or do we want to allow for lags, or do we want to look at certain frequencies? And these should ideally be guided by the questions that you're trying to ask about the data. But also some work has been done in terms of simulations. So for example, if you set up a network model and you assume some structure for the signals and the noise, and then you try to see which metric might best capture the underlying relationships you've set up. So there's work along those lines as well. Though we do have to also choose the assumptions that we put into our model about how the data are generated, or what numbers of nodes and what edges are we going to set up between them. And it's quite complex to know that we've covered any general case. So those are techniques for looking at undirected interactions, but what if we want to put an arrow between regions and say, does one region exert a causal influence on another region? So I'm not going to go into this in very much detail, but in Granger causality, which has been applied to fMRI data, causality, the notion of causality is based on temporal precedence. So basically, we're looking at the predictive value of a time course in one area and explaining a time course in another area, or the past history of one time course and explaining values in another region. And so, and in fMRI, there's been a lot of discussion, I think, and controversy over its use, and that stems partially from the fact that ultimately we'd like to look at neural causality, but we are observing our data through this lens of a hemodynamic response, which is generally not perfectly known between, like how that differs across different regions. And so, for example, what if the timing of region A in their neural activity happens before B, but then the hemodynamic response is more delayed? And this discussion is quite extensive, so I point you to these types of articles to learn more about that. And then there's sets of methods that focus on looking at usually a smaller number of nodes that you've pre-specified, and sometimes you pre-specify the connections as well, and it uses the joint set of activity across those nodes in order to try to best fit a model of causal interactions. And some of these fall under the umbrella of effective connectivity. If you are trying to directly model this in neural activity, then you need to set up a model that relates the neural activity to your hemodynamic observations. So in this last part, I wanted to discuss some challenges and considerations involved in the interpretation of functional connectivity data. And to start, I think many of you might agree with this message here, which is that functional connectivity can be a very powerful tool for uncovering many interesting phenomena in our data, but that it's also hard to clearly interpret these findings because there are many, there's ambiguities and it's not exactly clear even what our signals mean. So the data are noisy, for example. They're sensitive to modeling decisions and pre-processing decisions. When we look at resting state data, which is most commonly done in these situations, the spontaneous fluctuations that go into our time series could be a mixture of many and largely unknown processes. And there's not like a really clear mapping between functional connectivity with, for instance, structural connectivity or electrophysiological measures. But actually, this is all very exciting and open research directions that are being addressed in the field. One of the challenges in functional connectivity analysis of resting state data in particular is that distinguishing signal and noise is harder than in task-based studies, because here we don't have any model of our time course that tells us what we'd expect to see and to help us distinguish that signal and noise. And we can't really average across any event to try to boost that. And another thing is that, when we're doing functional connectivity and we're using these time courses that we extract from different areas, those, we're looking at the relationship between those, and each one contains some balance of signal and noise. And so rather than just comparing our data against a noiseless task regressor, we're actually comparing two things that are also may have some residual noise, and so this can create a danger of drawing the wrong conclusion about brain networks, especially if we don't carefully treat potential sources of artifacts. One of the well-known sources of artifact in our data is when participants move their head in the scanner, and it's been shown that this should be treated very carefully because even small amounts of head motion have been shown to impact connectivity measures. And one situation where this might be very problematic is that if there are systematic differences in head motion between two groups of subjects you're trying to study or across age groups, like because children may move more than adults, then this might cause spurious functional connectivity effects. And other things that impact functional connectivity are physiological processes like breathing and cardiac activity, and these actually impact fMRI signals in a few different ways. So if we consider breathing, there are certain types of fMRI artifacts that are kind of more time-locked to each breath, and it's what methods like retro-ICOR try to handle, if you've heard of that. But there's also slower natural changes in the depth of rate of breathing, which has been pointed out to also correlate with intrinsic activity, and this is right in the frequency band that we often try to study for resting state. And these directly modulate blood flow and oxygenation, and it can impact much of gray matter, and so it's a little bit harder and trickier to separate these kinds of artifacts from fMRI data using things like ICA. And you can also think about what impact this might have on network or connectivity analyses, which is that if you imagine placing seeds or nodes in different areas that are affected by these breathing effects in similar ways, then their connectivity might be inflated a little by that common effect. And one thing I would recommend is actually just always collecting physiological data if you can. I mean, maybe you might not use it immediately, but it usually comes in handy at some point, at least, useful information. And another consideration is that our results can be sensitive to pre-processing that we apply. So generally, it is hard to evaluate whether our pre-processing steps are doing the right thing because we don't really know what it should be in brain data. And so that can create a lot of debate and about exactly how we should process our data. And perhaps one of the most famous examples of this in resting state literature is this global signal question. So it was shown back in 2005 that default mode network may have negative correlations with attentional networks at resting state, which could be quite interesting and maybe reflect some kind of balance of internal and externally oriented processing. But the reason this became a little controversial was that because they use a step called global signal regression, which is basically where you take the average of all of your voxels across the brain, and you regress that out. And so it's kind of an aggressive way of trying to remove anything that might be common across our voxels, and was shown to mathematically introduce negative correlations in our data. But then how do we know if we should use global signal regression or not? This is still something that is kind of open. And because on the one hand, it might be too aggressive and artificially introduce negative correlations. But on the other hand, if we don't do that, it might leave too much common noise in the data and mask any real negative correlations that might be there. And so, but there's also other ways of handling this noise reduction, besides just regressing out a single global average. So there's the ICA-based methods, as we've already discussed, which try to reduce specific artifacts based on their spatial or temporal signatures. Or using things like tissue-based regressors, so you take some part of the brain where you don't expect neural activity, like the CSF, and you try to regress that out. Or to try to model specific sources of noise in our data, and then project those out. Project out specifically the ones that relate to the measurements of physiology, hoping that may provide something more interpretable. But this is all, there's still so little consensus on, the best ways for doing this, although there are certain studies that have provided some guidelines. And another thing I wanted to mention, though, is that changes in our functional connectivity might happen not just because of noise or artifacts, but also because of real neural sorts of variability. So like if you're looking at states of alertness or what the subject is thinking during the scan, that might actually change network relationships and patterns. And on the one hand, this can be kind of a confound, like if you're interested in quantifying connectivity or networks between groups, and you actually have more state-related variability than you get that swamps your between-group effect size. But on the other hand, this variability is actually something that's very useful to us that we can study, because it can reflect then these different principles of how the brain might reconfigure to support these different types of functions. And as you might hear about more later on, there's also been a growing number of studies on how these networks may change over time on rather short time scales from seconds to minutes. And so we can ask questions about whether these dynamic changes in network connectivity convey useful information that gives us a richer picture of what the brain is doing, or how these relate to certain ongoing internal states that might be going on. And so if we wanted to identify the sources that are driving variation in our fMRI signals, or to link functional connectivity to neural activity, there's several different approaches for doing that. And one of them involves actually trying to record complementary neural or behavioral indicators or physiological indicators during fMRI, such as EEG for measuring electrical activity, or other indicators that may signify emotion or alertness. And then we can essentially use those as a reference to try to identify which features in fMRI signals or networks might be linked with these different types of state changes. And as one example, EEG has been a gold standard for identifying sleep stages. And this study combined EEG together with fMRI to look at patterns in the fMRI functional connectivity that were associated with different sleep stages. And then they went a step further, which was actually to classify, see if you can classify sleep stages based on the fMRI patterns of connectivity in short time windows. So maybe be able to directly read that state information from our fMRI networks. And then one last method I'll measure here is, or I'll mention here is this idea of intersubject correlations, which has also been emerging as a promising way of trying to probe signal components or functional connectivity that's related to specific neural processes or states that's linked to a common stimulus. So the idea is that we present the same stimulus if you present the same thing to like each of your subjects, and usually people use a richer stimulus like a movie or a more complex narrative that they're listening to. And then if we correlate the activity of one person's brain to the activity of other people's brains, not necessarily in the same voxel, but almost like a seed-based thing where you can see, take the correlations with other regions in their brain. If you think about what this does, I mean, first of all, it seems like a really cool thing to do, but also it could also help us to isolate stimulus-specific changes in functional networks. So in other words, even though each individual subject is this mixture of stimulus signals, some intrinsic neural signal or non-neural signal that might be hard to separate out in that one individual, if we assume that the intrinsic signal and the non-neural signals are uncorrelated between subjects, but that the stimulus component, related component is shared, then that's kind of a way of kind of filtering out these parts and focusing mostly on the stimulus-related component. And so one of the applications that they looked at here was, well, they looked at this dynamic connectivity in the default mode network over time, and this was during a story, and showed that the connectivity of default mode network calculated in this intersubject way tracked these changes in the stimulus feature, specifically how well that section of the story was encoded in a person's memory. Oh, actually, one more thing to say, then we're done. So finally, I mean, we've mostly been talking about functional connectivity and these associations between brain regions. But it's important to keep in mind how these connectivity measures relate to kind of more first-order properties of our time series. So if we have things like a change in amplitude or a change in the spectral content of our signal or brief events, all of these might introduce, or they might cause us to see a difference in correlation. But is correlation really the best description of what's going on in our data, or would it rather be more meaningful to actually characterize this as a transient event? And so if we only look at associations and these kind of second-order descriptions, then we might miss out on some of the kind of the underlying things that are going on in our signal. And a lot of the methods development, I think, a lot of methods development has been driven by thinking a little outside the box of these connectivity measures and looking at other things in the data that may capture different aspects of brain function. So in summary, there's many different methods and metrics for inferring functional connectivity. And mainly I think it's important to be aware of how these work and what their trade-offs are, and would recommend staying as close to the data as possible, so looking at it a lot and trying to understand what's going on. There's many things that can introduce variability, and especially if you're looking at resting state data, because it's so unconstrained. And it might easily seem overwhelming to us, like how many possible things we might want to control for. But you know, I think, but I think there's like a lot of rich information to be found, and we just have to do our best of carefully controlling for confounds that we know about. And then there's still a lot of open questions surrounding how we interpret this data, and it could help as we move forward in better understanding the neural and physiological basis of our signals. So thanks a lot, and yeah.
for one quick question. All right, so the top-rated one from the online that's relevant here is, how does functional connectivity analysis change for higher bandwidth measurement modalities such as MEG?
Oh, how does the measurement or the kind of results, I guess?
Yeah, I guess how you would analyze it.
Yeah, well, I guess with MEG and measures where you can see faster phenomena in our data, like oscillations, there are more techniques that one can use, I think, that make more sense to apply there than maybe for fMRI. More techniques based on, for instance, phase locking or phase lags that may be sensitive to different, I guess, like connectivity between different regions or transfer of information between different regions. And also, actually, with fMRI, or with MEG, EEG, where we have longer numbers of time points, too, it often opens up for us more things we can do in the temporal domain because we have that temporal resolution and we have a long enough time series where we can more robustly quantify certain things than we can with fMRI, where we usually have shorter time series that are lower temporal resolution. I mean, even if we can push our TR to really short acquisitions, we're still physiologically limited by the vascular response. And so then there's a lot we cannot see about things like neural firing and all that.
Hey everyone, so as Alex mentioned, I do work on the Matrix 3 software package. I have tried to make this as an educational, as kind of software agnostic as I could, but that's my background. So this talk's going to be slightly different in structure. I'm kind of experimenting a little bit here. I decided when Alex and Andrew first asked me to do this presentation that there was simply no way that I was going to fit all of the details of quantifying streamlines tractography into a 40-minute session. So what I want to do instead is focus on the most kind of fundamental principles in the field and kind of try to give you the tools to think critically about the different mechanisms and methods and tools that you'll see in this field. So I'm going to start with a couple of points here just to clarify to you the way that I'm going to be thinking about the messages that I'm talking about today, and then I'm going to enumerate these four kind of what I've called fundamental requirements for performing tractography in a quantitative fashion. So as mentioned previously, we have the online poll system going. There's a dedicated poll for any kind of immediate questions on specific topics that I'll talk about, and of course there's the general Q&A for more general questions, and I'll pop up a couple of polls when I get to the relevant slides. So firstly, I want to talk about this way of thinking about the connectome and what we're actually quantifying. And basically when we talk about the connectome, prior to any actual analysis of the data thereof, what we're really doing is taking some pathway of interest, maybe defined by its two endpoints. We want to somehow reconstruct the connectivity between those 2 points, derive some measure of, ideally quantitative measure of connectivity, and then the connectome is simply repeating that experiment many times for many different grey matter regions. So just to demonstrate this visually, what I've got here is a brain parcellation where there are only, text my monitor is down if you're able to fix that. So what I've got here is a brain parcellation where all I've done is parcellate the actual lobes of the brain. So there's actually only 8 nodes in this parcellation, 4 per hemisphere. On the right-hand side, I've got this whole brain tractography reconstruction. And when we do structural connectome construction, we're combining this information together to produce a connectome matrix. But what does this actually represent? Well, if we look at the individual entries within this matrix representation, each of those entries corresponds to a specific pathway. So for instance, the 2 white squares highlighted here correspond to the right frontal lobe and the left frontal lobe, and the measure of connectivity we derive is based on the streamlines that connect those 2 regions. Correspondingly, we can look at the pathway between left frontal lobe and left temporal lobe, and so we get, for instance here, the arcuate fasciculus. We derive some measure of connectivity, we put that into our matrix, we repeat it for the next pathway. And obviously some pathways are going to be more or less connected than others. But the fundamental point that I want to get across here is that what we really want to think hard about is the mechanisms that we use to quantify the connectivity between region A and region B. Because once we've got that issue sorted, the question of connectome construction is just a matter of doing that many times over. So the reason I want to make this exceptionally clear is that in a lot of my talk I'll be talking about it may sound slightly disconnected from a connectome reconstruction or a connectome analysis, but I want to be very clear that all of the kind of little nuances and principles that I'm talking about are entirely relevant to connectome construction. I'm just kind of looking at the deeper level of what's going on. And finally, just for the sake of kind of showing a pretty picture, here you can see that same matrix representation, but obviously you have all of your different parcels along your rows and columns, and each one of these squares within your matrix representation is an individual white matter pathway. So are there any questions on that specific concept, either online or anyone can jump onto the microphones at any time? Nobody? Cool. I'm perfectly comprehensible. That's excellent. If I can get on from there. Okay, so next I want to talk briefly about streamlines tractography. Again, this is a clarification I want to make because the concept of tractography, there are a lot of different ways to do tractography, and if you look at the kind of state-of-the-art research in tractography, there's a lot of quite advanced ways of trying to do tractography. But when it comes to actual applications or people that are actually generating connectomes for future analysis, it's basically every single application is using what's called streamlines tractography. But it's important to recognise that that's kind of a subset of tractography. So this was famously introduced into the diffusion field in 1999. The interesting thing is in that 20 years, streamlines tractography really hasn't actually changed that much. The basic concept is still the same in that you have this kind of voxel field of estimated fibre orientations, and you're simply going to take incremental steps along that trajectory. So for each point, you look at the local you look at the local fibre orientations, you take a step in that direction, you look at the local fibre orientation at that next point, you take a step in that direction, and you repeat. It's actually exceptionally simple. And so I'm just opening up a new poll at the moment. Anyone who is more familiar with the field might want to make some suggestions of alternative types of tractography that don't use the streamlines paradigm. So personally, I like to break the streamlines algorithm into these four aspects. The first is what's referred to as streamline seeding. So this is the process of determining a location in three-dimensional space from which you are going to initiate a streamline for construction. The second step would be determining the appropriate orientations in which to propagate that streamline, and obviously this is done independently for each step as you generate that streamline. The third step would be streamline termination, so determining for a particular point in space whether the streamline should continue to be propagated or whether it should be terminated at that point. And finally, there's streamline criteria. So this could be a list of any length of criteria that you're going to impose on your streamlines reconstruction to decide for a given reconstructed streamline trajectory, do I want to keep this streamline or do I want to reject it? And this could be due to any of a wide range of criteria, from intrinsic tracking parameters like length restrictions to things like selecting only those streamlines that belong to a particular pathway. Now, just to kind of demonstrate the hidden complexity within here, in the MRTrix3 software, we have a command called trackgen. This is for generating tracks, i.e. doing streamlines tractography. And what I've done here is I've simply queried the software for the help page of this particular command. And what you can see here is the total printout of the help page of this command. So even though you can't actually see the underlying text, the main message that I want to convey here is that even though the fundamental paradigm of streamlines tractography is very simple, there's actually a lot of kind of latent variables, a lot of different dials that you can tweak. And every single one of these little dials is going to influence the result that you get from streamlines tractography. Now, obviously, I'm not going to go through every single one of these with you. I simply want attendees to be aware of the breadth of the total number of parameters that are available to you, and the fact that for probably the majority of these, there is no single correct answer. There's no correct parameter to use. Obviously, in software implementations, we will have sensible defaults or the like, but these shouldn't be considered as being set in stone or being perfectly optimised. Okay, so do we actually get any results on the poll for types of streamlines that are not streamlines based? So I'll rattle through these quickly. There's bedpost X. Bedpost X is actually a diffusion model, not a streamlines algorithm. So in the FSL package, bedpost X is what fits the diffusion model, and prob track X is the one that does tractography. So bedpost X is actually the former of the two. Both Gibbs and global I would put into the kind of set of what's referred to as global tractography. So streamlines tractography is often referred to as local tractography because as the streamlines propagated, it's only the local fibre orientations that are taken into account, whereas global tractography tries to fit the entire brain reconstruction as a single process. So it's a lot more complicated. If I can actually get this to progress. Here we go. I had listed a few others there, but unfortunately it's skipping over them. So other ones to be aware of would be geodesic tractography, which is based on fixing the endpoints and then looking for a specific path between those 2 points. There are also graph-based tractography approaches whereby instead of considering a graph of brain regions and having connections between the brain regions, you consider every image voxel to be a node within a graph, and you look for pathways through that graph, which represents a three-dimensional tractography trajectory through your image. So, yeah, without going into a huge amount of details on each of those, just understand that streamlines tractography is a subset of tractography. But, yeah, sometimes people can use the two interchangeably, which gets confusing. Okay, so as I mentioned at the start, I didn't want to try to go through all of the different techniques or softwares or the like that are available for streamlines tractography. So what I'm really going to focus on in the second part of this talk is what I've over time in my own experience kind of recognised as the most fundamental underlying principles in terms of if you want to use streamlines tractography to quantify connectivity, and particularly if you're looking to do so in a way that does actually have genuine quantitative properties, then there's this set of what I've identified as 4 kind of underlying fundamental principles that you need to address. And so these are kind of sit at a higher level than the details of any particular software or any particular algorithm. They're simply things that I would encourage you to think very critically about whenever you are investigating which software you want to use, or if you're reading a manuscript that's used a particular technique or that's proposing a particular technique, to really think hard about whether or not these principles are satisfied. So here's the list back on my overview page. They are trajectories, attribution, reconstruction density, and quantification. So we're going to step through these one at a time, and I'll give the opportunity for questions after each individual point, and then there'll be time for more general questions at the end of the presentation. So starting with trajectories, the basic principle here is that within the brain there are white matter fascicles. Those fascicles pass through some kind of spatial trajectory through the white matter. With tractography, we're trying to reconstruct those trajectories. Now, if our trajectories that we digitally reconstruct are in no way representative of the actual underlying biological fibre bundles, then we're wasting our time. So this is kind of the very first principle that we need to be thinking about. And this is influenced by a couple of separate steps, so I've broken this point into two. The first would be estimating the local fibre orientations, and the second would be reconstructing trajectories based on those estimated orientations. So focusing in on the first of these points, so the role of the what's called the diffusion model is to take the diffusion MRI signal in each image voxel and try to reconstruct from that one or some set of fibre trajectories that tell you about the orientations of the underlying fibre bundles in that region of the brain. Now, it's important to recognise that with streamlined tractography, we're relying on having an accurate estimate of fibre orientation at every single step in order to reconstruct the entire trajectory accurately. And so if at any point along this streamlined reconstruction process from left to right, if at any point along that path we get just one incorrect fibre orientation, the entire trajectory is then going to be completely wrong. So it's exceptionally important to get the most robust and accurate measurements of fibre orientation that we can. And so I've created another poll there asking about whether people are aware of some of the different diffusion models that are available in the field that can potentially be used for the purposes of tractography. So what do we actually need? What parameters do we need from our diffusion model in order to perform this reconstruction? Well, ideally, we want accurate estimates of the fibre orientation within each image voxel. Probably the more famous one is this colloquially referred to issue of crossing fibres. Basically, what this means is that within a single image voxel, it's possible biologically that there may be multiple fibre bundles crossing at various angles. And so therefore, in order to be able to reconstruct those fibre bundles accurately, it's exceptionally important that the diffusion model that we use for tractography be able to properly resolve the fact that there are multiple different fibre orientations within that voxel, but also accurately estimate the individual orientations of those individual fibre bundles. It's also possible within an image voxel that the underlying white matter fibres may not be perfectly coherent, but there may be some degree of kind of orientation spread. And so ideally we would like to be able to encapsulate this. From the perspective of digital reconstruction, it would be handy to have some knowledge of the uncertainty associated with our measurements, because then we might be able to estimate from that the kind of uncertainty in our connectome reconstructions. And ideally, we don't want to be constrained by the fit of the diffusion model in each image voxel, because if the streamline position is in between two image voxels, then ideally we want the orientation that we follow to be in between the orientations of the fibres in those two voxels. So here I've just quickly tabulated some of the kind of most famous tracking algorithms. So I'm hoping a lot of you will have heard of the diffusion tensor model or diffusion tensor imaging or DTI. Most famously, this gets a big fat cross in the crossing fibres column because the tensor can only represent a single fibre orientation in each image voxel. And there have been experiments that have shown that the fraction of white matter that actually contains crossing fibres is very high. So it's a very high proportion of the white matter where the diffusion tensor model is inadequate. Probably the most commonly used software for tractography would be the FSL software. The underlying diffusion model is called ball and stick or ball and sticks. So this does deal with the crossing fibres problem because in each image voxel it can potentially fit multiple what they call sticks, where those sticks refer to the fibre orientations. However, it doesn't model any presence of orientation dispersion within those. With the software that I work on myself called MRTRIX3, the underlying diffusion model we use is called constrained spherical deconvolution, or just CSD for short. So with this one, we do get somewhat of an estimate of fibre orientation dispersion. We can perform interpolation reasonably well. Again, I don't want to torture you with all of the intricate details of these. The main point that I want to convey is that whenever you see a particular diffusion model or tractography algorithm, you want to think critically about whether or not these kind of needs are satisfied. So did we manage to get any responses on different diffusion models? We did. So there's ball, yeah, it probably falls under ball and sticks. There's a method called commit. That's probably something I'll talk about later in the session because that's a slightly different concept. There's one here called IFOD. So IFOD refers specifically to a tractography algorithm rather than a diffusion model. Probabilistic is a specific class of tractography, so that's again something I'll talk about later. Okay, so I think we've got that covered, and unfortunately it likes to hang on me whenever I bring up a poll. It's not progressing for you either. So if I can... Sorry, is it possible to just progress by one slide at your end? Here we go. Sorry, it's now jumped ahead a whole lot because I was clicking like mad. Okay, so the second aspect of this issue is once you've got the fibre orientations, you then need to follow them in order to reconstruct the streamline trajectory. And as I showed previously with the command help page, that depends on a very large number of different parameters. The most basic of which would be, okay, at each point, how much is the streamline allowed to turn? Ideally, you want the streamline to keep going in the same direction rather than doing a 180-degree turn and going back the other way. And there's probably some kind of distance between adjacent vertices. A few other things to consider. Obviously, we've spoken briefly about the model. There's the order of the integration. So fundamentally, Streamline's tractography, you can think of it mathematically as an integration technique. You want to use a technique that performs that integration accurately and doesn't accumulate numerical errors. Alex mentioned previously the distinction between deterministic and probabilistic tractography, so that's shown on the right here. For deterministic tractography, if I generate 10 streamlines from the same starting location, I get the exact same streamline 10 times, whereas with a probabilistic algorithm, I get 10 unique trajectories because for each one, it's making independent samples of the underlying uncertainty in the orientation. I briefly mentioned the step size and the angle, and there's a few other things that I'll talk about in subsequent sessions. So are there any more general questions regarding this first point about Streamline's trajectories? No? Cool, I'll move on. And we can always catch up on stuff at the end of the talk. Excellent, there we go. Okay, so point number 2 is what I've called attribution. The language here might be slightly cryptic. Essentially what I'm talking about is that if you've got 2 grey matter regions and you want to reconstruct the pathway corresponding to the connection between those, the way in which you assign individual streamlines to those grey matter parcels is important. This is a detail that a lot of people kind of skip over, but I want to focus on it a bit more here. So the first challenge here is in the actual termination of the streamlines. And this comes back to what we actually measure with the diffusion signal, in that it doesn't really give us a good, while it's good at telling us the orientations of the fibres, it's not very good at telling us where they start and stop. So for instance, here in this little diagram, what I've got is 4 different fibre configurations, 4 different voxels, yet the diffusion signal in each of these is identical. Yet in the bottom 2 examples, you can see the fibres terminate, whereas in the top one, they don't. So figuring out where to stop a streamline based on the diffusion signal alone is exceptionally difficult. So there's a few different ways that people have tried to do this in the past. You can constrain the angle, how much the streamline's allowed to turn. If it tries to turn too sharply, you terminate. You can have some measure of anisotropy from your diffusion model. You can derive some kind of image mask. You can constrain the maximum length. What tends to be more strongly advocated these days is to actually exploit multimodal image information. So it's quite common in an MRI experiment to acquire a high-resolution, high anatomical contrast image, like a T1 weighted image. What we can do is use that image to explicitly segment the image into different types of tissue, and then use that tissue information to inform the streamlines process. So this is a demonstration of a method I developed that's called ACT, or anatomically constrained tractography. Basically, the point of this image is that in the middle image, you can see all the streamlines that are coloured. Each of these yellow dots is a streamline termination, and you can see that streamlines are terminating absolutely everywhere, which, for the purposes of connectome construction, is kind of problematic because it makes it very difficult to figure out for a streamline which grey matter parcels should I be assigned to. Whereas if you make use of this tissue information, don't allow streamlines to terminate in the white matter, only allow them to terminate near the grey matter, then the whole process becomes a lot better posed and your connectome construction will be a lot more robust. However, this does mean that any kind of geometric distortions that are present in your diffusion image acquisition, those need to be explicitly corrected accurately, because otherwise your distorted diffusion information and your non-distorted T1 weighted image information won't align properly with one another. So step 2 of this attribution point is on assigning streamlines. So I alluded to this briefly. In the challenge of taking streamlines and assigning them to grey matter parcels to generate a connectome, there's actually some kind of explicit process going on in there, in that actual assignment of streamlines to parcels. And the way that you do this can actually have a very large influence on the connectome that you get out as a result. So for instance, you could simply look for those grey matter parcels that the streamline intersects. Now this can be problematic. So for instance, this streamline here in the left-hand side image, it's passing through the grey parcel and the yellow parcel, but it's kind of arguable as to whether it actually should be, because it's not actually travelling to and terminating at those regions, it's kind of erroneously passing through them. There would also be an issue here whereby the fact that this streamline passes through 3 different parcels, not 2, means this streamline would actually contribute to more than one edge in the connectome. So how you actually think about that biologically or physically is a bit weird. What we tend to advocate is some kind of heuristic to make this process as robust as you can. So all we do is at each streamline termination, we kind of do a little search around the local spatial neighbourhood and find whichever node is closest to it. So I personally think, I don't know how many people would agree with me, that for this particular streamline, the most appropriate outcome is for this streamline termination to be assigned to the orange parcel, and for this end of the streamline to be attributed to the blue parcel. Probably the more robust way to do this would be to interact directly with a surface-based representation of the cortex. There are some softwares to do this, but obviously that comes with its own large number of caveats. So are there any questions that are specific to this point number 2 on streamlines attribution? Yeah. Yeah, so for instance, with the ACT method, the fundamental assumption we're making there is that axons do not synapse in the white matter, which is known. They synapse in the grey matter. So therefore, we don't allow streamlines to terminate in the white matter. We force them to terminate, in our case, at the interface between grey matter and white matter. So at least that biologically makes sense. Obviously, we're ignoring cortical depth of the light or the like. And obviously, if point number 1, trajectories, isn't satisfied, then you may have intersected with the wrong point of the cortex. But I think that the fact that you know, having used that technique or a similar technique, that you are actually terminating at cortex, personally I think that satisfies that to the best of our technical capability. Yes. Yeh et al. Neuroimage 2017, I believe. Okay, so we'll jump to number 3, reconstruction density. This one's hopefully fairly accessible. Basically, you have to generate enough streamlines so that your connectome construction is reproducible. So a couple of kind of thought experiments. Say you choose a number of streamlines, you generate your connectome. Now let's say you take the same image data and you regenerate your streamlines, and then you regenerate your connectome. If those two connectomes are different, then you've got an experimental problem. Ideally, you want to be generating enough streamlines so that if you simply repeat the experiment with the exact same image data, you should get more or less the same result. The question then becomes, all right, how many streamlines do you need? Obviously, if I say I have a connectome with 100 parcels in it, if I generate 100 streamlines, chances are I'm going to get a different connectome every time. If I generate 1 billion streamlines, chances are if I then generate another 1 billion streamlines, I'm going to get the same result, but I'm going to be waiting a very long time. So this is a balance that needs to be determined in the experimental design. And fundamentally, the answer to this question is going to depend on exactly what you are quantifying for each individual pathway. So for instance, I pulled this image from a thesis where here they're specifically interested in the spatial volume that is occupied by a particular white matter pathway. And so when you've generated only a few streamlines, you kind of get an underestimate of the volume because the streamlines algorithm hasn't had the opportunity to fill out all of the space where that pathway may actually exist. And so as you generate more and more streamlines from left to right here, you gradually get a more complete estimate of the total volume occupied by that pathway. Now, obviously, there's the caveat that you may also start introducing more false positives and therefore you may overestimate your volume. That's kind of a more nuanced discussion about the quantification of pathway volume. Again, thinking about the more fundamental concept is if you were generating a tiny number of streamlines, so if you were down in the left-hand side here, not only would you be underestimating your volume, but it would probably be slightly unpredictable as to what volume you would get. And if you redid the experiment, you'd keep getting different answers. So you want to be up towards the right-hand side of this graph. Exactly how far you should go depends on the particular experiment that you're doing. So I've posed this question, and apologies, I'll just need to activate it. If you were... oh, somebody else has activated it for me. So what I've posed here is you've got 3 different parcellation schemes. In one of them, you've only got 10 nodes. In one, you've got 100 nodes. And in one, you've got 1,000 nodes. And my question to you is, how many streamlines do you think would be an adequate number of streamlines to generate in each of these cases? You know, what do you think would be a good choice for your experimental design? And again, I would refer back to the kind of statement that I made at the start of this section, in that whatever it is that you're quantifying, you need to have enough streamlines so that that quantity that you derive is reproducible if you repeat the experiment. Apologies, I probably didn't quite give enough time to actually give some numbers here. So let's see what people thought. 100, 1,000 and 10,000. I suspect if you've got 1,000 nodes and you make 10,000 streamlines, that's going to be far too sparse. 1,000, 10,000, 2,000, sorry, 1,000, 10,000, 20,000, probably a bit small again. Somebody suggested 10 million. They're probably familiar with my work because that's my golden number. There's another 10 million. So one way I would suggest at least thinking about this, even if it's not a golden rule, is that the number of unique edges in the connectome scales quadratically with the number of nodes. So if you've got 10 nodes, you've got about 50 unique edges. If you've got 100 nodes, you've got about 5,000 unique edges. If you've got 1,000 nodes, you've got, 500,000 unique edges. So if I propose a basic heuristic and say, okay, I want to have an average of 1,000 streamlines per edge in the connectome. Well, okay, if I've got 10 nodes, I'm aiming for about 45,000 streamlines. But by the time I get up to this, 1,000 nodes in my parcellation, suddenly I'm asking you to generate 500 million streamlines. Now that's a large number. That's going to take a lot of computation time. That may well be an overestimate, depends on other nuanced details. The point here is that the number of streamlines that you actually need in order for your experiment to be robust does in fact accelerate very quickly once you start going to much larger numbers of nodes. Are there any other more general questions about that topic specifically? Is that a hand? No, it's not. No hands? Cool. All right, 5 minutes left. Get to the final point. This is the fun one. Quantification. In each edge of the connectome, we're quantifying something. And it has something to do with connectivity, inverted commas. What we're actually quantifying needs to be both robust and it needs to mean something biologically. So again, fundamentally, what are we doing? What are we actually trying to quantify? What are we trying to capture in our connectome representation that we're then going to do some higher level analysis with? So a couple of suggestions that have been already raised by Alex in the first talk. You could simply take the number of streamlines. The trouble then is that the number of streamlines is known to not be quantitative. Yes, it probably tells you something, like it's probably vaguely proportional to some connectivity thing, but it's also affected by a lot of other things. And so there are a lot of weird biases that you would have in your quantities that you don't really want. You can do something like take the fractional anisotropy from the diffusion model, which is supposed to tell you something about integrity in exceptionally large inverted commas. The trouble here is that you're inheriting all of the weaknesses of the diffusion tensor model, you're then inheriting into the quantity that you're using in your connectome. So it's not fantastic. And then if you're going to take those quantities and you get a connectome matrix and then you do some complex analysis of your connectome matrix properties, what does that mean? The whole thing kind of becomes almost too abstract to be of any use. So this is when I say our, my kind of collaborators and people who we work on the matrix software with, is what we fundamentally want in a structural connectome when we represent our brain as a graph, is we want something akin to bandwidth. We want to say, okay, these two nodes, there's a lot of bandwidth, there's a lot of capacity for information to flow between them. These two nodes over here, they're not so much connected. It's not possible to get a lot of information quickly from A to B. And what we essentially use as a proxy for this kind of bandwidth is the fibre intracellular cross-sectional area. Now, it's a bit of a mouthful, but I'll show it visually and hopefully it will make more sense. So what I've got here is two regions of interest on the left and the right here in the kind of transparent grey, and we've got a bunch of fibres connecting them that I've just drawn as cylinders. Now, if we look at a cross-section of this bundle, so this square in the bottom left here is looking at the what you see when you project that cross-sectional slice onto the screen. And so these circles here correspond to the area within each of those cylinders. And so what we're deriving as our quantitative property is to sum up the cross-sectional areas of these bundles, and that sum hopefully gives you something that is at least in some way proportional to the capacity of this biological system to transfer information from this block on the left to this block on the right. Now, obviously, there are a lot of caveats here. There's different axon diameters, there's myelination, there's X, Y, Z. A lot of these things are not very accessible to us in diffusion imaging, and the encapsulation of such as really at the state-of-the-art development stage of diffusion MRI, not really yet applicable for use. But what we're proposing is that this measure is probably the best we can get at this point in time. So what we actually want is estimates of this cross-sectional area between each pair of grey matter regions. What we have from our diffusion model is estimates of fibre orientations and densities. So this is another method that I created called SIF2, and basically the premise here is that if you have your brain, this square down the very bottom left is a map of fibre density. Now, if you generate the same map, but if you look at the density of streamlines, these two images don't match one another. And this is one of the fundamental reasons why we classically say that streamline count can't be used in a quantitative way. What this SIF2 method does is essentially modulate the cross-sectional areas of the fibre bundles that are estimated by each individual streamline so that the local fibre densities give a reasonable estimate of the local fibre densities that were estimated by the diffusion model. And by doing so, that gives us some confidence that our estimates of cross-sectional area connectivity between grey matter regions is at least to some extent biologically accurate and relevant. So I'll skip by that final slide. There's a few different ways that you can contact me with regards to particularly the Matrix 3 software package. We've got a workshop later in the year if anyone wants to learn how to use some of these methods. And just briefly, I've also got a poster on Wednesday talking about an encapsulated workflow to apply a lot of these techniques in a fully automated fashion. Unfortunately, we've really got very little time for any further questions, but I'm hoping I've caught things as they came. So if there are any big outstanding questions, we can do them now, otherwise we've probably got to jump to the next talk.
Maybe we'll just do one quick one that's been voted up a couple of times. So how does ACT account for the possibility of pathologic fibre interruption, such as demyelination or stroke? And yeah, another question, how do you deal with brain lesions in white matter?
Yeah, okay, so those two are both the same. So the answer to this is specific to ACT. Within the tissue parcellation, it's possible to manually define, to manually delineate tissues that you are classifying as somehow pathological. And in those regions, basically what you're saying is that you don't know how the streamlines algorithm should behave. And what specifically the ACT method will actually do in those regions alone is to disable ACT. So the tracking algorithm kind of defaults back to its original behaviour of only using diffusion information.
Cool. All right, so last talk before lunch, you're probably going to get sick of the Australian accents, but it's my pleasure to introduce Andrew Zalesky from the University of Melbourne, and he's going to be talking about network statistics and null models for inferences on networks.
So I'd just like to begin by thanking Alex for doing all the heavy lifting and putting together this education workshop this year, as well as all the speakers for participating. So my presentation is going to consist of 3 parts. I'm going to begin with a brief discussion of network thresholding. I'll then move on to null models. And in the third part, I'll wrap up on network statistics and comparing networks. My slides are available for download, so if you go to connectome.org.au/OHBM19.pdf, you can download all of the slides that I'll present today. So to begin with, network thresholding. What is thresholding? Well, thresholding is really taking, once you've mapped a connectome, is to remove potentially spurious or weak connections from the connectome that you've mapped. And here I've shown a case where we can successively reduce the connection density by going to 20%, 10%, and all the way down to 5%. It should be emphasised that thresholding is not an essential step. It's not necessary to perform thresholding, but some of the reasons to perform thresholding are that it can improve the clarity of connectome visualization. So rather than having a bird's nest, we can really pinpoint the strongest, the most important connections comprising a connectome. Thresholding can assist in removing spurious or weak connections. It can emphasize certain topological properties. And some other points there for thresholding are also that it's recently been shown that thresholding can improve the reproducibility of connectomes. And it can also ease the storage and computational burden of very large connectomes, so connectomes computed at the voxel or the vertex level. So here's more of a topological view of thresholding. So here we have a toy graph. The graph comprises of 3 types of connections. I've labelled them strong, moderate and weak. And so the weakest are the thinnest edges, the thinnest lines, the strongest are the thickest lines here. And so initially, the graph has an inherent connection density of 26%. And the first step that we can do, we can perhaps say, well, the weak connections are perhaps spurious, and so we can remove those from the graph. So here you'll see that the connection between this node has gone, and this node has gone here, and this results in a connection density of 20%. We can then go a step further and perhaps remove both the weak and the moderate connections. So our resulting network only comprises the strongest connections. What you can see happen here is that one of the nodes has become disconnected or fragmented from the network, and this can cause problems when we go on and compute our network measures or do further analyses on the network. It's also probably not biologically plausible. So there's good reason to ensure that our graph remains fully connected when we perform thresholding. An inherent property to thresholding is the concept of connection density. It's a really simple concept, and it really dictates or it determines how strongly our thresholding or how stringent our thresholding is. And the connection density is simply the total number of connections that remain in our network after we've performed thresholding, divided by the total possible number of connections in the network. And here's a really simple example of a toy network with 9 nodes. It comprises 16 connections. We can assume perhaps that's after thresholding. And so the connection density is 16 on 36 or 44%. So to compute the total possible number of connections in a network, you compute that using the formula here. It's simply n, where n denotes the total number of nodes, times n minus 1. So each node can connect to all of its possible neighbours except for itself. That accounts for the minus 1. And we divide by 2 because it's an undirected network. Once we've thresholded our graph, so this is a step where we've performed thresholding. The black cells here, the black elements denote connections that have been removed as a consequence of the thresholding process. A further step that we can perform is binarization. And so that's shown here in the bottommost graph. And when we binarize, we're only interested in the presence or absence of a connection. So the white cells represent connections that are present, black are absent connections. One of the reasons for doing binarization is that perhaps we don't have strong confidence in the weights estimated with our connectome generation process, whether that be with tractography, so whether our streamline counts, the variation or the dynamic range across streamline counts is biologically plausible. To take away our the assumptions that we make on that, we can binarize and therefore not make those assumptions on the streamline count. So there are 2 very broad classes of thresholding, and here I want to present those to you in terms of, once again, a very simple toy graph. The graph, the numerals that I've indicated on the graph edges, these could, for example, correspond to streamline counts. One way to threshold the graph is using an approach called density-based thresholding. And this is where we set a prescribed density, so in this case, for example, 20%, and we rank our edges from weakest to strongest, and then we successively remove the weakest edges until we arrive at our prescribed connection density. And so in this case, we are left with 3 connections. So in this case, the graph is actually fragmented. So perhaps that density threshold is too low. The other way to threshold a graph is with weight-based thresholding. And in this case, rather than prescribing a connection density, we prescribe a weight threshold. So we put a threshold on our streamline count or our functional connectivity. So in this case, we say 3 streamlines. So any connection that comprises fewer than 3 streamlines is removed from our graph, and this is the resulting graph. Now, the key point here is that for any weight-based threshold, so if you tell me a weight-based threshold, I can give you an equivalent or the corresponding density threshold to achieve that graph. So there's a one-to-one correspondence between weight and density-based thresholding. So you might say, then what's the point? What's the point of actually making a distinction between these two methods? If there's a one-to-one correspondence, so if you tell me a weight, I can give you a density, or if you tell me a density, I can give you a weight. Why are you creating this distinction between these two methods? Well, the reason is that when we do group-level analyses, or when we're thresholding across a set of subjects, this distinction becomes apparent. And this becomes apparent even if we're only looking at 2 subjects. So in these 2 subjects, subject 1 overall has a greater proportion of strong connections, as indicated by the thick edges, compared to subject 2. So in this case, if we do weight-based thresholding, and we say, well, we're going to eliminate both moderate and weak edges, we see that once in the thresholded graph of subject 1, the thresholded graph comprises more connections than the thresholded graph of subject 2. And this is due to the fact that there is a difference in connectivity strength between these 2 subjects. So if we were then to go on and do network analyses and compare a particular network measure, whether that be path length or clustering between these 2 subjects, we would find a difference, a statistical difference, but that difference is most likely can most likely be ascribed to this difference in the number of edges between the 2 subjects. So what can we do to solve this? Well, you would probably say, let's do connection density based, let's do thresholding based on connection density. And so sure, by definition, if we threshold on connection density, what we would do, we would then add in a few more connections into the thresholded graph of subject 2, and so then they're both matched in number of edges, in the number of edges, and therefore that potential confound, that difference in the number of edges between subject 1 and subject 2 is removed. But we've then introduced a potential additional confound, and that is that what we've really done, we've lowered the bar for subject 2, and these weak and moderate edges that we've added for subject 2 are potentially spurious. Potentially they're false positives. And these false positives can dramatically impact on our subsequent network analyses and network measures that are computed on the graph. So on one hand, just to recap, on one hand, density-based thresholding ensures that our subjects are matched in terms of the number of edges, but we need to potentially lower the bar and add spurious connections, whereas weight-based thresholding has the confound of having different number of edges across subjects. And so that's borne out in this example here. So this is an example in schizophrenia. So in schizophrenia, individuals with schizophrenia generally have lower functional connectivity than healthy comparison individuals. And so with weight-based thresholding, if we use a weight-based threshold of 0.2, so any connection less than 0.2 is set to 0, whereas the remaining connections above that threshold are kept, we see that the patient has a connection density of 53%. In contrast, the healthy control has a much higher density of 75%, and this can confound any subsequent network analyses. However, if we do density-based thresholding, by definition, we solve this issue, so we can impose a density threshold of 50%, but to do that, we've had to lower the bar for the patient. So we've had to admit weaker connections into our thresholded graph. In particular, we've had to lower the bar to a minimum connection weight of 0.31 in the patient, whereas the minimum threshold in the control is 0.42. An important question is, how do we choose our density threshold, or how do we choose our weight threshold? And it's really an arbitrary choice. There's no good reason. We don't know what the underlying connection density of the brain is. And so one way to get around this is to consider a range of connection densities. So we can threshold our network across a range of densities, compute our network measure, or repeat our analysis for each of those thresholds. And then, for example, we can plot the network measure as a function of the connection density. And to derive a summary statistic, we can consider the area under the curve. And this gives us a single value to characterize that network measure across a range of connection densities or across a range of weight thresholds. And so this takes away, to a certain extent, the contingency on one specific weight or density threshold. This is another example of another thresholding method. So I should first say that density and weight-based thresholding are by far the most common methods for thresholding a connectome, but there are other methods that have been proposed. This is a consensus-based approach. The basic idea here is to keep connections that are consistently found across a group of individuals. The difficulty with this is it's often that the spurious connections are also consistently found across individuals, and therefore they can be preserved with this method. There's also the MST, an MST-based thresholding approach, which guarantees that the network remains connected. So it specifically avoids, or it's specifically developed to combat that issue of nodes fragmenting, disconnecting from the network. I won't go into the mechanics of that here, but if you want to take a look at the slides and have a look in more detail. There are also local thresholding methods. So everything I've described so far would be considered a global thresholding method. We take one threshold and apply it to the network as a whole, and all edges in the network, all connections are subjected to that global threshold. Another class of thresholding methods are local methods, and this is where, rather than applying a global threshold, we actually look within the neighbourhood of a given connection and see how strong that connection is within its local neighbourhood, within its locale. And this can potentially allow us to recover finer structures in the network, finer detailed structures. But I would say that these methods are not widely used in connectomics. So as I said, some of the more advanced methods are the MST. There's also a local method called the disparity filter. I won't go into the details of that here. Take a look at the papers by Serrano and Foti. There's also multi-resolution methods, which to a certain extent take into account the issue of the arbitrariness in the threshold choice. So to summarise, I've presented global methods for thresholding, weight and density-based thresholds, and given you a bit of an idea of the confounds that are associated with those two methods. There's the consensus-based and MST, which are more niche. I've also briefly touched on local methods for thresholding. Once you've thresholded a graph, and once again I need to emphasise, thresholding is not essential. You can perform weighted analyses. It's not crucial to threshold a graph. But if you do decide to threshold, you can then take one further step and binarise the graph, in which case we're only interested in whether a connection exists or not. And to take away the arbitrariness of a threshold choice, we can consider things such as the area under the curve, where we repeat our network analysis over a range of thresholds. So I just wanted to point out these two recent papers. So one of the questions you might say, should we threshold or not? And in this first paper, the basic argument, this is actually, the basic argument made here is that, well, thresholding doesn't really impact network measures to a large extent, and given that, why should we add this step to our processing pipeline if it doesn't have a dramatic impact? That's the first paper. The second paper here really takes an opposing view, and this is a preprint that suggests that thresholding structural networks can help to improve the strength of association with behavioural measures and age. This was done in the UK Biobank. So this kind of presents two opposing views, one against and one for thresholding, particularly in structural networks. So I'm going to move on to null models. Are there any questions on thresholding? Okay. Null models. So what to motivate the rationale for, or the need for a null model, I want to consider the network measure of path length. So Bratislav will go into more details about path length in the afternoon, but for now it'll suffice to say path length is really just the number of synapses that need to be traversed in a neuronal network. So in this case, we have a direct connection. Therefore, the path length, as denoted by L, is 1. We only traverse one synapse. In this case here, we traverse 3 synapses, and therefore the path length is 3. And so the question that I want to ask is 3 long? Is that a particularly long path length? Is it average? Or is it short? To answer that question, we need some kind of benchmark. We need a reference to determine whether this value of 3 is necessarily long, short, or average. So one way to do this, we could consider a small network, and we say we're going to benchmark this to a random network, and we're going to say our random network is going to comprise 9 nodes and 15 connections. And there are analytical formulas that can be used to compute the path length in a random graph. If you're thinking, what is a random graph? Well, a random graph is just where we have a group of nodes and we randomly draw connections between pairs of nodes until we reach a desired connection density. And so the formula for path length in a random graph is just the number of nodes squared divided by 2 times the number of edges. And so for a network of 9 nodes and 15 connections, this turns out to be 2.7. And so we can say for a network of this size, for a small network, a path length of 3 is pretty typical, right? It's not, maybe it's a little bit longer than average, but it's fairly typical. In contrast, if we consider a bigger network, so now if we have 27 nodes and 45 connections, and we can compute that same formula again, remember this is the analytical formula for path length in a random graph, it turns out that the path length on average is much longer. It's a path length of 8. And so if we find that our path length in our real connectome is 3, we can say, well, this is pretty unusual. A path length of 3 in our real connectome is quite short. And so this is telling us that there's something perhaps optimized in the connectome that's allowed us to compute, that's given rise to this short path length. So the point I want to make here is that we need some kind of reference or benchmark to determine whether a network measure, such as path length, is necessarily long or short. So this kind of reference would be given by a null network. A null network is also referred to as a surrogate network or a benchmark network. And the 2 properties of a null network are that, first of all, the property that we're interested in, so path length in the example that I've just given you, should be annulled, or it should be taken out of that null network. And often that's quite hard to do. The second property is that the null network should be matched to our real connectome in all other possible properties. And so the simplest type of null network is a random network. As I said before, this is just by taking nodes and drawing edges between those nodes at random. So what I've shown here is kind of the space of possible null networks or possible reference networks that you can use to benchmark. In the outermost circle, I've considered all possible networks. So arbitrary number of nodes, arbitrary connection density. This is probably not a very useful null model. It's not sufficiently constrained. So then we can say, well, what about we match our null network to the number of nodes in our real connectome? So we only sample our null networks from those networks that have the same number of nodes as our empirical connectome. That's generated here with the blue. Then we can say, well, we can constrain our null model a little bit more. We can also constrain it such that it comprises the same number of edges. And what you can see here, gradually as we come closer, we're going to actually converge. If we add more and more constraints to our null model, we're going to converge to our actual connectome, to our real network. So what's most typically done in connectomics is that we match our null network so that it has the same number of nodes, the same number of edges, and it's matched to the node degree of our real connectome. And that can be achieved using an approach called Maslov-Snepen rewiring, which I'll touch on in a moment. We can also add, and what has received recent attention is that we can also match our connectome, our null networks, to the geometry of our real connectome. So by geometry, I actually mean the length of connections. So we only draw, or our null network, the distance of connections in our null network is matched to the distance of connections in our real network. And this further constrains our space from which we sample null networks. and it can determine what effect the spatial embedding or the geometry of the connectome has on network measures. So what do we do once we've done this, once we've determined exactly what our null network should comprise, what constraints it should keep or not keep, how can we then use this? And so what's done, we typically sample random networks from our space. So remember the pink space here is where we match both the nodes, the number of edges, and the node degree. So we keep sampling random networks from this space that preserve these three properties. And we can compute the average path length. So if we're interested in path length, we can compute the average path length across those null networks, across those random graphs, and compare that to the path length in our real network. And so this could be done just by taking a ratio. And so if this ratio is very large, it would suggest that the path length in our real network is much longer than what we would expect as a matter of chance. In contrast, if it's small, the interpretation would be the opposite. We can also use this to compute a p-value. So if we take the proportion of random networks for which the path length is shorter than our actual connectome, this can allow us to do hypothesis testing relative to that particular null. So as I said, in connectomics, the most common null is where we preserve the degree, the number of edges, and the number of nodes. And to do that, this can be done quite straightforwardly with a technique called Maslov-Snepen rewiring. And so with Maslov-Snepen rewiring, we randomly choose 4 edges in our empirical connectome, such that 2 of those nodes are connected, so UV and IJ, such that we have this specific motif. And then we rewire this. So rather than U being connected to V, we connect U to I, and we connect V to J, and then we put this motif back into the original place in the connectome. And we keep doing this again and again. We randomly select a motif such as this 4-node motif. We rewire it as such, and then we put that back in. And by iteratively doing this again and again, we randomize the network, and crucially, we preserve node degree, we preserve the number of edges in the network, and quite obviously, we preserve the number of nodes. We're not adding any nodes in doing this process. To convince yourself that we preserve node degree, you can see here that the degree of this is still 1, 1, and 1. And so once we embed this back in the network, its degree of the 4 nodes will be preserved. So here's a little example of Maslov-Snepen rewiring in the macaque and the mouse connectome. This is some work from Bassett from a couple of years ago. So this, on the left here, we see the original, the empirical connectomes, and we can then rewire these using Maslov-Snepen rewiring. So this is what I've shown here is just one sample from Maslov-Snepen rewiring. In practice, what we would do, we would sample from this space multiple times and therefore generate multiple null networks such as this. And as I've mentioned, we can then compute, for example, the clustering, we can compute path length and normalize them. So we have our path length in our original macaque connectome. We can take the average path length in our rewired networks and take the ratio. And in this case, we see that the clustering is quite high. And this tells us that the clustering in the macaque connectome is significantly greater than what we would expect due to chance, or what we would expect under our null model. In contrast, the path length is just about the same, so the ratio is about 1. So it's saying that the path length in the macaque connectome is pretty much the same as what we would expect under our null network. And so this gives us really the conditions to diagnose a small world network, and we'll see that more in the afternoon, how that can be done. And this is the same for the mouse connectome. Once again, we can generate multiple null networks, take the ratio of our measure, whether that be clustering or path length in our real network, divided by the average across that measure in the null networks. And as I've seen, as consistent with the macaque, we see that clustering is greater, path length is approximately same, and these are the conditions that give rise to a small world network. So there are also advanced null networks. You might wonder, how do we deal with Maslov-Snepen rewiring in the case of negative edges. And so the typical instantiation of the algorithm doesn't allow for negative edges, but there's been some nice work that was done quite a while ago now by Mika Rubinov and Olaf that extended this algorithm to the case of edges with negative weights, or in other words, signed networks, as well as directed and weighted networks. In fact, for directed networks, the traditional algorithm works fine. For weighted networks, once again, we need to make some modifications. As I've already mentioned, there is increasing work that's incorporating the actual spatial embedding of the connectome into the null network. So it's increasingly recognized that the connectome is a spatially embedded network, and so by preserving the geometry of the connectome in our null network, we can test how much of to what extent a network measure is related to the connectome spatial embedding. Finally, another approach is using generative networks. These are not really null networks per se, but this is another approach that can be used to benchmark or reference null networks, and Rick may talk about that in the afternoon. So before I move on, are there any questions on null networks? No? I'm just looking through the questions that have been posted online. Perhaps we can get to those on in the question and answer session after this. So I'll move on to comparing connectomes. So once we've mapped our connectome, so you can consider a very simple scenario where we've mapped a connectome for a group of healthy controls, we've mapped the connectome for a group of patients, and we want to compare, we want to understand what features of the connectome differ between the patient and the control group. Or alternatively, you might be interested in understanding what behavioural measures are associated, or what connectome attributes are associated with a particular behavioural or cognitive measure. Or maybe you might be interested in, you've administered a particular intervention and you want to understand what connectome attributes are mediated by that intervention. So how would you go about doing this kind of network comparison, and what kind of attributes of the connectome would you compare between groups, or would you test for associations with a behaviour or an intervention? So I like to think of network comparison and network statistics across three levels. Now, as a caveat, I should say this doesn't include multivariate comparisons or multivariate approaches for statistical comparison. I'll get to those in a moment. But at the bottom here, we have global comparison. This is the case where we characterize each individual's connectome with a single value, with a single network measure. So for example, this could be the average path length across the entire connectome. And we could then compare the path length between patients and controls, or we could test whether the path length correlates with a certain behavioural measure, and we can use our standard statistical toolbox to undertake this kind of inference. We only have one value associated with each individual. So this is really easy to do. The disadvantage of this approach is that it doesn't give us any specificity. It doesn't tell us what part of the connectome, what node or what edge is actually driving that association, or that's driving that between group difference. So to allow that, to improve specificity, we can then move on to what I've shown here in yellow. We can perform comparison at the level of individual nodes. And so there are node-based measures, as we'll see in the afternoon, such as clustering, or for example, we can compute the path length at the level of individual nodes, and then undertake those same comparisons. The difficulty with this is that we end up increasing the number of multiple comparisons. So n here corresponds to the number of nodes. So while we have better specificity, we can actually localize the effect to a given node, we also have to deal with the problem of an increased number of multiple comparisons. And if we take this to the extreme, we can also ask what specific connections, rather than looking at the nodes, what are the connections that are most strongly associated with the behaviour or that differ between a patient and a control group. This substantially increases the number of multiple comparisons, so the number of multiple comparisons are then in the order of n squared. So this raises statistical challenges in dealing with this, but it also gives us really good topological specificity in the sense that we can isolate, we can pinpoint an effect to a specific connection in our network. So I'm only going to focus on connection level comparison here. This, I guess, is the most challenging area for network statistics. One way to do this is using mass univariate testing. And so this is where we can fit a general linear model, or any kind of model that we wish. In this case, I've indicated a general linear model to each edge of our connectome. And so this endows every edge with a test statistic and a p-value, an uncorrected p-value. And so with a general linear model, we can implement a t-test, an f-test, an over and co-regression, and so on. So this could be, for example, testing differences between patients and controls, or with testing an association between, for example, the streamline count or the functional connectivity and a behavioural measure. And so this test statistic gives us the strength of that association. How do we then deal with the multiple comparisons problem? The fact that we've done this across thousands, potentially tens of thousands of edges. So one way to do this is to use generic methods, such as the false discovery rate or Bonferroni. Perhaps Bonferroni is probably too strict in this situation. I'm not going to go into the mechanics of the FDR. Take a look at my slide, which gives you an example of FDR. But what I do want to say is that what these generic methods, such as FDR and Bonferroni, is that they ignore the domain-specific knowledge, the particular configuration of effects that we see in a network. So in a network, it's most likely that the effects form clusters or subnetworks. We don't get an effect at some edge, you know, an edge up here and another one down here, and as I've shown here on the left. This is quite unlikely, particularly in neurological disorders, where misfolded proteins typically spread through the connectome in a sequential propagation, but also in psychiatric disorders, as well as in behaviours. It's more likely that effects are circumscribed to networks, to what we call here subnetworks or clusters. And so we can use this domain-specific knowledge to improve the power to detect between-group differences or associations with behaviour. One way to do that is with the network-based statistic. To briefly give you an overview of how that works, essentially what we do, once we have a test statistic at every edge, we can convert that test statistic to a p-value or to an effect size, and we say, what's our minimal meaningful effect size? And we say any edge, any connection that's below that minimum meaningful effect size, we throw away. And then we have a set of super threshold edges, so those edges that are above that minimum meaningful effect size. And the idea then is to identify clusters or subnetworks in those super threshold edges, in those edges that have a sufficiently high effect size. And in this example here, I've shown a network that comprises a frontotemporal network that comprises 5 connections. So then the question is, what's the probability that we would observe such a network just as a matter of chance? What's the probability that we'd see a subnetwork with 5 connections as a matter of chance? So to ascribe a family-wise error-corrected p-value to that network, we can use permutation testing. I won't go into the mechanics of that, but just to give you a flavour of what we do, it's essentially permuting subjects. So this is in the case of a patient control design. This can be generalised to other designs as well. Permute subjects and see, once we randomise subjects, what's the probability that the largest subnetwork that we find exceeds the size of the network that we found in our real connectome, and that was 5. And we can see here that in most of the cases, well, we found one permutation where it was of size 10, but the other ones it was always smaller. And in this way, we can build up an empirical null distribution for the largest connected component, and we can see that our observed component of 5 is quite rare. Not many of these permutations resulted in a connected component that was of size 5 or greater. And in fact, it turns out we can compute a family-wise error-corrected p-value just by counting, or it's actually given by the proportion of permutations for which the maximum subnetwork was greater than or equal to 5. And so this way we can ascribe a p-value to a connected component. Here's some examples of this approach being applied to disease connectomics, so identifying subnetworks that differ between individuals with a certain disorder, whether it be psychiatric or neurological, and comparing them to healthy comparison subjects. So the key idea here is that we're using domain-specific knowledge, the fact that these effects are generally circumscribed to a cluster or a subnetwork, to help us improve statistical power. So rather than testing or rejecting the null hypothesis at an individual edge, we're rejecting across a group of connections. I haven't considered multivariate approaches for performing network inference, and this is a growing area of research. The advantage of, or the disadvantage of what I've just presented is that we treat every individual edge independently. So we fit our model, our generality model, to each edge without consideration of what's happening at another edge. With multivariate inference, we're really interested in looking at complex patterns that span multiple edges, multiple subnetworks, and testing how these relate to behavior or between group differences. So I've given an example of some multivariate methods there, increasingly CCA and partial least squares. There's also regression, sparse regression-based approaches, and there are network-specific ways to perform regularization. There's MDMR, multivariate distance matrix regression. There's also an increasing role of machine learning techniques that have been applied to network prediction and network inference. I guess the disadvantage of these is that they don't, or it's sometimes difficult to identify what are the specific connectome attributes that are driving the prediction. So I'm going to wrap up there. Here's some software to perform our network inference on connectomes. There's some work that we've done, some of our own work in NBS as well as other toolboxes. I should emphasize that the functions that you might have seen outlined in red to perform network thresholding and to develop network null models are available in the brain connectivity toolbox. So this is available in MATLAB. I think there's also a Python version of this toolbox. So if you want to implement any of the functions that I've discussed on your own data, this is a freely available toolbox to perform network statistics as well as pre-processing and computation of network measures. So I'm going to stop there. I've just about run out of time. We now have a question and answer session. So I think we can open this up to make it general to all of the speakers from this morning. But perhaps we can take a few specific questions from my talk before we open it up.
I just asked this morning's speakers to come up.
Mm-hmm. So do the speakers from this morning want to? So are there any questions specifically to network statistics? Yep. You might want to come to the microphone. It's probably difficult. There's two microphones at both corners of the...
So I was wondering, what about longitudinal design of your study? Will it help in any way in doing all those comparisons between the two groups?
Yeah.
Because then your baseline is your own subject.
Yeah, so of course with a longitudinal design you have a repeated measure. It's a within group design which affords you greater statistical power. So in the methods that I've discussed, there are some things that need to be done to account for that. So the way that permutation is performed needs to be constrained to account for the fact that your design is a within subject, or it's a repeated measures design, and so the design matrix needs to be also set up to account for that. So certainly within subject's design, a longitudinal data can offer advantages. I guess that's the advantages that it offers are not something specific to connectome analysis. That would also apply to more traditional neuroimaging analysis based on voxels as opposed to dealing with connections in a graph.
Might take this opportunity to ask you the burning question that's hit number one with a bullet, given you were just talking about thresholding. So is it true that it's wrong to threshold functional connectomes and we'd rather work with full graphs?
So I think the short answer to that is no. No, it's not wrong to threshold a functional network. However, it's also perfectly fine to use weighted graph analysis on a functional network, and that would not require any kind of thresholding. So I don't think that there's a clear right or wrong answer. I think to say that it's absolutely wrong to threshold a functional graph is not correct. I mean, one of the advantages of thresholding, for example, would be if you were doing statistical comparison, it would reduce the number of multiple comparisons. So if you had some a priori information that an effect would not be present between a particular pair of regions, then why not remove that? Why not remove that connection from your statistical analysis?
Should we just work through?
All right.
Some of them have been asked. So, perhaps for Catie, is there any way to solve the indirect correlation issue?
That's a really hard question. I don't know if there's a way to solve it. I don't know if anyone else has any thoughts on that, but I guess, yeah, from just looking at fMRI data or just looking at our data alone, it's always a little bit hard to tell. I guess increasingly people are trying to do more experiments, I guess, in animals, for example, if you can actually try to do some kind of causal intervention to try to like better tease apart which areas might be directly connected or which might be indirectly connected, or by looking at patient populations where you know that there is a lesion or some severing of connectivity, and trying to understand what patterns of connectivity persist or not. But if anyone else has anything to add.
Yeah, I would maybe ask to what extent it really is an issue, because, you know, it depends on what you want. If you want to really only look at structural connections, then maybe you need to use a combination of structural and functional connectivity methods to look at that. But if it contains, I mean, the brain is such a highly connected network that indirect connections might well contain a lot of information and might well be really interesting from a behavioral perspective. So is it an issue or not is a question to ask as well.
Yeah, I think the reasonable answers. I mean, the only thing I would add is that obviously there are methods for estimating effective connectivity. DCM is one. There are others out there with slightly different takes on the underlying biophysical models. Then, of course, there's partial correlations, regularized partial correlations, and so on. Each of these measures have got their own free parameters and assumptions, so there's no kind of silver bullet to try to just focus on direct interactions. And so I think it depends on the level of your interest, of inference. Correlation coefficient's probably a good first pass, but then if you really want to drill down, perturbational methods or trying to use some kind of effective connectivity model would be a nice complement to that.
So I think there's an interesting question here on alternative methods of connectivity estimation. It specifically relates to using the gyrification index for connectivity and comparing that between a patient group. So I think we've specifically focused on diffusion MRI-based connectivity. Yeah, so the question is, what do you think about using jackknife-based estimation to extract subject-specific values from a correlation matrix computed across a group of subjects, for example, using gyrification? So I think this refers to structural covariance. So how would this compare to, for example, diffusion MRI-based connectivity? What would be, is there a correspondence?
I can't say that I fully follow the kind of proposal that's being made here. The only alternative technique I'm familiar with is you look for correlations in cortical thicknesses between different regions and you use that to infer connectivity, but in that case you only get one measure for the entire group. So no, I'd have to think about that proposal in a bit more time.
So I think one comment on that would be that there is recent work that's used, so rather than using a simple measure, a single measure like gyrification, if we can combine a group of measures like gyrification and cortical thickness, we can then derive an individual connectome for a subject. So basically you're looking at correlations across a group of measures. So that could be cortical thickness, perhaps a diffusion-based measure, So I think there's some work by Jacob Seelitz that's looked at that.
Yeah. I mean, I do worry sometimes there's a bit of a danger in simply trying to figure out a quantity that you can get for each edge in order to produce a matrix so that you can do a matrix analysis. Whereas I would personally think about, okay, what are the actual parameters that are being quantified in your connectome matrix? Because if those don't make sense in a graph-based representation, then any subsequent analysis of the graph data are not going to make particular intuitive sense either.
Okay.
Okay, so how about, aren't we potentially getting rid of important information by density thresholding?
Yeah, look, if we threshold too stringently, we can. So I think it ultimately boils down to what is a genuine connection, what's a real connection, and what is a spurious connection. So we know it's very clear that tractography can result in connections between regions that are genuinely not connected. And so if we include those connections in our connectome, these can have a dramatic effect on the computation of graph measures. In fact, in recent work, there's some work that Alex, I, and others have done. We've shown that actually the false positives have twice as much detrimental impact on network measures than false negatives. And so one of the conclusions of that work is that we should threshold stringently to remove, or to put more emphasis on removing false positives as opposed to keeping the true connections, the true positives. But of course, if we threshold too stringently, there's always the risk of removing genuine connections.
I do think we need to make a kind of terminology clarification here. It's something we run into with the manuscript that Andrew showed on one of his slides, is we quite commonly get into this talk of thresholding, but there is actually a difference between applying a threshold to remove weak connections and then binarising the result. And I think those two need to be thought of separately and explained separately. Because, for instance, the result with respect to consequences of false positives versus false negatives is specifically in the context of binary connectome analysis.
Yeah, no, I agree. I mean, the distinction needs to be made. So I think for binary connectome analysis, by definition, there is thresholding, right? There has to be, otherwise
there's thresholding, but there's also binarisation.
Yes.
Part of the reason why I think that distinction is important is, for instance, the manuscript I was an author on that advocated against thresholding was specifically looking at thresholding but not binarising. So it was removing weak connections but still retaining connection strengths. And in that case, the empirical behaviour is very different, because in a binary connectome, removing one connection or adding one erroneous connection can have a very large influence. Whereas, for instance, in the experiments we did in that manuscript, the distribution of connection strengths that we get in our structural connectome constructions span about 5 orders of magnitude. So if you're removing these very weak connections that have only got a few streamlines, but then there are other connections in the network that have got 100,000 streamlines, removing those weak connections basically doesn't have any effect. But then if you start ramping it up further and further to the point where the thresholding does have an effect, you then have to ask yourself, well, are the differences that I'm seeing differences in the metric that I'm looking at, or are they differences that are being imposed by how the thresholding is influencing different subjects differentially? So it's a weird problem, but I do think that distinction between simply removing weak connections and binarising needs to be exceptionally explicit.
Thanks, Rob. So I think we have a question.
Yeah, thanks. Could you comment, please, on the potential for quantifying connectivity within the reticular activation system in the brainstem, where you don't have fascicles and you've got synapses in what looks like white matter?
I think that's a very interesting question, because we know that those have widespread connections to so much of the brain and so important for regulating brain states. I think one challenge, well, so fMRI data, I guess, would be most suited for this since we have, more spatial resolution, but still those areas are very tricky in terms of like them being tiny regions, very noisy. And so I think the acquisition and denoising plays a very important role in being able to get good signal to noise ratio and know exactly where you are in this kind of complicated territory. And then, yeah, hopefully if we can do that, then I think there's more potential for studying directly the connections with other regions.
Probably the structural connectivity answer to that would be that if we can't see them on a T1, then we can't even segment them, so we'd never have any hope of quantifying connectivity using diffusion with or without use of the anatomical information. But fundamentally, that's an anatomical resolution and contrast issue, it's not an issue that's specific to streamlines connectivity.
Any other questions from the floor?
Another online one. So back to the correlation issue. In your opinion, would it make sense to combine partial correlation and regular correlation, for example, by averaging the resulting coefficient or in some other sophisticated manner to ensure that the findings are independent of the specific method? Has this been done?
Well, okay, I might have to think about that more, but I guess, I don't know, I mean, they might give different results, so you might not, in terms of if you're looking for like confirmational evidence from like one, like looking at both of those, so then it's hard to say like which one you use, something that is more along the lines of what you're trying to study. Okay.
I think perhaps you could take a hybrid approach, right, and actually regularize. So rather than partial correlation is at one extreme, full correlation is another. We can use sparse approaches where we can
balance the cost function between the two of them somehow, end up somewhere in the middle. Yeah, I mean that's an intriguing idea actually. I don't maybe interesting to try and see what happens, but I don't have a good intuition about what that would give us.
I mean, I would be very careful about just averaging things together. I think that's I would advise against, because that's going to potentially be very difficult to interpret because you don't know what's driving things. I think the more you regularize, the more, I mean, that's essentially systematically moving between fully unregularized partial correlation and moving towards full correlation. So the more you regularize, the more it's going to look like that. So that's kind of a way of being in between the two. It can be useful to look at both, I think, because, trying to understand why something shows up in a full correlation and not in a partial correlation might give you some intuition as to what's happening.
So I might just close on one last somewhat controversial question, which has been getting a few votes. So do we agree that at large scale, it's meaningless to use structural connectivity as a ground truth for functional connectivity? Discuss.
I mean, I guess I started by saying that it's structural nodes should not be used for functional connectivity. So I hope we can all agree on that using the AAL Atlas or the Harvard-Oxford Atlas is probably a little outdated, and we can do better at that level for functional connectivity using functionally defined regions.
I'd agree that based on Sokal and gyral landmarks is problematic, but defining parcellation, say, based on microstructure, which will be increasingly possible with things like the Big Brain Atlas, as the analyses of that are getting more realistic. Then, you have anatomically defined nodes of the brain, and then you can ask questions about the function. Naturally, if you do a parcellation based on function, you get something different, and that's a slightly different question. So I think that they're just addressing slightly different questions.
I think based on the wording of the question,
connectivity, right?
It's not talking so much about the parcellation itself. It's talking more about the quantification of tractography-based connectivity being some kind of ground truth for what should be coming out of functional connectivity. So in that case, I would say it kind of depends on what you mean by ground truth. You certainly can't assume any kind of trivial relationship within the strength of one particular edge between two modalities. If you think more critically about what's actually arising to the functional connectivity measurement, you've got your biological brain, you have some kind of fixed underlying wiring, and you then have stimuli and natural biological processes that are leading to certain types of activity in each node, and your functional connectivity is simply some measure of correlation of that. So the functional connectivity quantification is kind of a long way removed from the measure of structural connectivity. So as I say, there's not that one-to-one mapping, but what I would say is there's a lot of methods that are looking at, okay, if I have a brain, if I set these parameters related to how I think the grey matter behaves, and then if I use my structural connectivity as the kind of fixed underlying wiring, can I then determine the parameters within that model for which, when I then run a simulation, the functional connectivity that I measure within that simulation is the same. And I think there's a lot of merit in that kind of approach, because it's really getting to the underlying biological mechanisms as well as the mechanisms of what we're measuring.
Yeah, to add a little, so yeah, like structural connectivity is probably like a important constraint probably on what kinds of functional connectivity patterns the brain can support. But like, as we know, even from like looking at, we know that dynamic connectivity can be modulated, and perhaps depending on different brain states. And so there may be more like, I guess, traffic on the network in some areas than another in different parts of time, depending on what's going on. And so that might not relate directly to like the strength of structural connectivity, but be more like the states that are supported on top of that architecture.
I think, yeah, there is a relationship, but you don't want to impose a simplistic relationship.
I mean, it might be a little bit controversial, but the way I like to think of it is, there's an underlying structure, and those connections have weights, and they, I guess, are constraint on the overall capacity of the connection. Then on top of that, you have effective connectivity. So if we had perfect knowledge of the brain, we could define all the nodes perfectly accurately. We knew all the nodes, we knew all the connections. Then ideally, we would have a perfect map of effective connectivity. And they're going to be directed connections that are going to be completely constrained by the anatomy. But we have imperfect information, and doing effective connectivity is hard, so we use proxies like functional connectivity. And depending on the measure we use, we start to get all these indirect interactions and so on. And so that creates some ambiguities. Does that mean all those indirect connections are false or not informative? No, they're probably picking up polysynaptic connections that probably have behavioural relevance and so on, but it just reflects a kind of limitation of the methods and the measures that we have. But I'm sure other people have different opinions.
I think it probably wraps up the session. So we have our lunch break now, so we'll reconvene at 1 p.m.